All right, good morning, everyone. We're going to go ahead and get started, try and keep us on time today. We've got a lot of great speakers, so definitely going to want to keep all the time we can for all of them in their presentation. So welcome to our second annual Update in Schizophrenia. Hard to believe that we were here just about a year ago, and time flew. Certainly not much going on in the world during this past year. So we'll hopefully have a great discussion, and particularly when we get to the Q&A. So I'm going to just move through our slides, and particularly want to give my colleague here, Mike Levert, a chance to welcome you all as well. We're really excited to be presenting not only on an important topic, but we've done a couple of other highlights this year. And so trying to continue to update all of us in terms of what's going on in the clinical field as well as in the science. Certainly want to thank Mr. Levert and the Levert family for their support, their generous support in helping us get this kicked off last year, and to be able to continue to provide this important content to all of you. So I'll invite Michael to come on up and say a few words of welcome as well.
Good morning. I'm Michael Levert. First of all, thank you for the speakers that are here today. Thank you to Endeavor Health and Dr. Soka and his colleagues for getting this all set up and organizing it all. Thank you to our sponsors, for Federal Savings Bank and Greenway Metal, for helping sponsor this symposium. Thanks to my colleagues, my two daughters, Catherine Owine and Elizabeth Levert, and my wife of 47 years as of yesterday, Anna Levert. And my two siblings that came today, Jean and Greg, thank you for coming. A special thanks to Dr. Carl Wallstrom, I think is attending via WebEx, and Dr. Zach Bowa, who's part of Endeavor Health, who's also attending via WebEx. I'm Michael Levert. I'm the founder of the Lauren Marie Levert Foundation to Cure Schizophrenia. We started the foundation a few years ago in order to honor my oldest daughter, Lauren Marie Levert, who contracted schizophrenia in 2003, we believe, and she died in 2010 after 7 years of battling the disease. We're not sure if she actually contracted the disease in 2003, but that's when we first started noticing symptoms. From what I understand, for the more we've gotten involved in the area, it seems like there's a lot earlier signs than the ones that we probably noticed. And that's good that there are earlier signs, because the earlier schizophrenia is treated, the more successful the outcomes are. Okay. Today I stand before you to discuss a transformative frontier in mental health research, the use of artificial intelligence to uncover the causes of schizophrenia and pave the way towards effective treatments and hopefully ultimately a cure. Schizophrenia is a profound and complex mental health condition that affects millions of lives worldwide, disrupting thoughts, emotions, and behaviors. For too long, its causes have remained elusive, and its treatments, while helpful for some, have fallen short of addressing the full spectrum of its challenges. But now, hopefully with the power of artificial intelligence, we stand on the cusp of a new era in understanding and combating this disorder. Schizophrenia is not just one puzzle, but a mosaic of mysteries. Its symptoms, hallucinations, delusions, disorganized thinking, and emotional withdrawal vary widely from person to person. Its origins are equally complex involving a tangled interplay of genetics, brain chemistry, environmental factors, and life experiences. Traditional research methods, while invaluable, have struggled to untangle this web due to the sheer volume of data and the intricate patterns hidden within it. This is where artificial intelligence steps in, offering tools that can process vast data sets, identify patterns invisible to the human eye, and accelerate discoveries at an unprecedented pace. Imagine AI as a tireless, brilliant collaborator. It can analyze genetic data from thousands of individuals to pinpoint specific gene variants linked to schizophrenia risk. By sifting through millions of DNA sequences, AI has already helped researchers identify over 100 genetic regions associated with the disorder. These findings are not just numbers, they are clues to the biological underpinnings of schizophrenia, guiding us towards targeted therapies that address the root causes rather than just the symptoms. Beyond genetics, AI is revolutionizing how we study the brain itself. Advanced imaging techniques like functional MRIs generate enormous amounts of data about brain activity. AI algorithms can analyze those scans to detect subtle differences in brain structure and connectivity between those with schizophrenia and those without. For example, machine learning models have identified patterns of reduced connectivity in certain brain networks that may contribute to symptoms like disorganized thinking. These insights could lead to biomarkers, measurable indicators of the disorder, that enable earlier diagnosis and personalized treatment plans. But AI's potential goes beyond understanding causes. It's also transforming the search for cures. Developing new medications is a slow, costly process, often taking decades and billions of dollars. AI is changing that by simulating how thousands of chemical compounds interact with brain receptors. AI can predict which molecules are most likely to alleviate symptoms without the harmful side effects. This approach, known in the silicon drug discovery, or is known as in silico drug discovery, is already speeding up the development of new treatments for other diseases and holds immense promise for schizophrenia. Moreover, AI is helping us rethink treatment itself by analyzing data from electronic health records, wearable devices, and even smartphone apps. AI can track symptoms in real time, predict relapses, and suggest tailored interventions. Imagine a world where a person with schizophrenia receives a gentle nudge from an AI-powered app to seek support before a crisis occurs. This kind of proactive, personalized care could transform lives, reducing hospitalizations and empowering individuals to manage their condition with greater confidence. Of course, the road ahead is not without challenges. AI is a tool, not a magic wand. We must ensure that the data used to train these systems is diverse and representative, so that the findings apply to all populations, not just a select few. We must also navigate ethical questions, such as protecting patient privacy and ensuring AI-driven insights are used responsibly. Collaboration between scientists, ethicists, and individuals with lived experiences of schizophrenia is essential to guide this with compassion and integrity. As we look to the future, let us embrace the potential of AI to eliminate the shadows of schizophrenia. With every genetic clue uncovered, every brain pattern decoded, and every new treatment tested, we move closer to a world where schizophrenia is no longer a lifelong burden, but a condition we can prevent, manage, or even cure. It's not a distant dream, it's a possibility within our reach, powered by the ingenuity of AI and unwavering commitment of researchers, advocates, and communities like ours. So let us unite in this mission. Let us support the scientists harnessing AI to unravel the causes of schizophrenia. Let us amplify the voices of those living with the condition, ensuring their experiences shape this work. And let us hold fast to the hope. Together, we can transform the lives of millions, bringing light to those who've been left in the dark. Thank you.
All right, powerful words. Thank you, Michael, for kicking us off here. So with that, I'm going to transition to our first speaker. Oh, thanks. Dr. Ed Cook. Ed is the Earl Bain Professor of Psychiatry and Director of the Center for Neurodevelopmental Disorders and Associate Head for Clinical Affairs at the University of Illinois Chicago Department of Psychiatry. You can see his distinguished bio here. He attended University of Texas Medical Branch at Galveston and completed both adult and child and adolescent training at the University of Chicago, and then moved over to University of Illinois in 2005. We're really excited to have him speak today on a very important topic, translating the complex genetics of autism spectrum disorder. So I'll kick it over to Dr. Cook, who's online, by the way.
Very much. Hear me okay?
Yeah.
Great. Really appreciated the introduction and pleased to be here. And I know it's Autism and a Genetics Symposium, but I'll bring the links together along the way. No commercial disclosures to report. We'll start with ADHD genetic epidemiology. This is why so many scientists moved into autism genetics. Some of these numbers may have changed, but the point is risk to siblings is 20%, 20-fold higher than the population. After tumor affecting the SIBSHIP, it's 1/3 to 1/2 recurrence risk for next children born. Obviously much higher, but what really drove everyone into the field that were not interested in autism or mental health in general was this finding of 90% identical twins. If one had it, the other one did. There's nowhere a much smaller increased risk to fraternal twins. And the genetic contribution estimates actually center more around 90%. This was published in Science in the 90s and drove a lot of our colleagues from more general genetics into the field of autism. And similar data drive geneticists into the study of schizophrenia as well. These are genetic etiology groups. This is also true in schizophrenia. De novo, meaning neither parent had the variant, but the person with the disorder does. Rare de novo events are predicted in genetically lethal disorders. Now, genetically lethal means a disorder that affects the chance of having children. This is true of both autism and schizophrenia. And the individual effect of these variants is often very large. I think one example people probably be most familiar with would be something like Down syndrome, where neither parent has an extra chromosome 21, but the child does. The aggregate of all of this is about 10% of ASD. That doesn't mean every patient that I see will have one that we know is the case. And I'll get into that a little bit later, but these are very, very high risk factors, often sufficient to cause the disorder. Then the inherited variants, the rare ones have large effect, and these are often examples would be recessive conditions. The common variants, This has been studied throughout psychiatry and autism as well as schizophrenia. There aren't as many hits in autism, partly because the data sets are larger in schizophrenia, and also because there's more rare de novo variation in autism. But now this is interesting contrast, because any of these individual common variants is not the same as some of the rare de novo variants, which is sufficient to cause the illness. But on the other hand, these common variants, their aggregate effect is large in ASD. It contributes to about half of ASD risk and applies to most of the patients. So this is a genetic model of autism. It's interesting, it's remained the same, although when I first presented it, the 2 to 5 overlapping circles, which you could think of as, we thought of as individual variants, where if you had 5 of them, you would have ASD. Now each of those circles would be hundreds or thousands of common variants. And it's really a question of whether someone had enough of them to go over the threshold of the disorder. And then there are less complex cases, which are more the rare de novo events that I told you. Some are inherited. And I'm going to emphasize those quite a bit more, because I think they're better clues to biology for the individual. They often are contributing to differences, for example, of additional medical challenges that someone might have in addition to autism. So we'll start with chromosomal disorders, insertions, deletions. This is an additional copy or deleted portion, not of a whole chromosome, but of a part of a chromosome. And I've listed here some of the most common deletions of a piece of chromosome. 7q11, Williams syndrome, 15q11, q13. You're going to hear a lot about that part of chromosome 15 today. So deletions there lead to Prader-Willi syndrome, Angelman syndrome, and duplications are going to be quite relevant to ASD, which is what I'll say in addition to autism. 16p11.2, 22q11, which you're going to hear about from me, but is actually a stronger risk factor for schizophrenia as well. And then 22q13 deletion. Of those common deletion or insertion regions, there's autism risk in many of them. The 7q11, not deletion, but duplication. Interestingly enough, the 16p11.2 deletion. ASD risk, I'm going to tell you the duplication also has schizophrenia risk. And the 22q11 deletion, we're going to talk about as an overlapping schizophrenia and autism risk. So I mentioned chromosome 15. It's spent quite a bit of time on. So this is sort of a diagram of chromosome 15, and there's not much happening on the small arm or P arm. The Q arm, the 1,1 to 1,3 region, which is actually the first subband of the first band. So it's really actually the first 3 subbands of the chromosome that has genes that matter. This would be the normal. If it's deleted on the chromosome that came from the mother, it leads to Angelman syndrome, missing this piece from the mother, severe or profound intellectual disability, general cognitive problems, and ASD risk, which is actually high in Angelman syndrome. Eternal deletions, Prader-Willi deletions, missing this from the father, exact same deletion region, but the origin matters. Mild ID, or maybe not intellectual disability, but other learning problems without general cognitive impairment, often related to severe obesity, and also obsessive-compulsive difficulties beyond the obesity issues. So those are deletions. Just think of this region as missing. I'm going to focus a lot on the duplications because of their relevance to autism spectrum disorder. This second grouping here is an increase of this region, a duplication within the chromosome. So the total number of chromosomes would be 46, would not have changed. Actually, technological advances were mentioned very appropriately earlier. These were actually very hard to detect with chromosome technology, and I'll show you an example of how modern genetic methods are enabling much of what we're now discovering. But this would be an insertion within the chromosome. I'm actually showing it on the maternal side because, as you'll see, those are the ones that greatly increase autism risk. There can also be deletions on the chromosome that came from the father. We think that has much less of these three possibilities, the two different kinds of deletions, the two different kinds of duplications. The duplication from the father has little or no effect in contrast. And then there's another grouping of an extra piece of a chromosome. And these are actually, they're easy to detect that there's an extra piece of a chromosome on the traditional sort of gray banded chromosome analysis. But you really do have to make sure that this Q11, Q13 material is there, because if not, it's actually not going to cause difficulty. So this, I just still see this patient regularly. About, I guess I saw her first in about 88, meaning 1988. And this is an example where she has this extra chromosome. This is a kind of banding where it's the centromere being, or staining where it's the centromere being stained, and she has, they're called pseudodicentric. They're basically the DNA of centromeres, but they're not acting as two centromeres. And this is actually from the larger chromosome prep showing the two normal chromosome 15s and this extra piece. And this is the first woman that I saw that really led me into an interest in this area. Severe intellectual disability, epilepsy, which has fortunately been relatively easy to control, repetitive behaviors, anxiety about lack of order. When she came to me, she had self-injury, mood lability, and she came to me on haloperidol, and had she remained on that amount of haloperidol for the last 30 plus years, she would likely have the side effect of tardive dyskinesia. But she had a good response to carbamazepine and an SSRI, removal of the self-injury and other behavioral problems, and she's actually doing quite well. This is an open label case study. This does seem to be more helpful for the disorder, and may be an example of, probably is an example of precision medicine, where knowing the genetic condition helps to define the treatment. Other things that you think that one might think would be more helpful, even for the epilepsy, aren't as helpful as carbamazepine. I like to point people to this parent group. One of the advantages of identifying this syndrome of duplications in this region is the families get together. I would talk with one of the parents, and they'll affectionately refer to other dupers that they've met at conferences. Knowing that there are similar problems is very helpful. Sharing at their meetings things that work or don't work is also very helpful. This is actually a picture. I don't show you the gray banded picture, but I'll just make the point that we identified this in a laboratory using microsatellite markers. And when we sent off for the gray banding, we got the report back that there wasn't a duplication, but this was one of the first advances, which is metaphase FISH, where it's most obvious in this C figure that instead of one band here, there's actually two bands of this region. So even with metaphase fish, it wasn't as obvious on each of these figures. This led to interphase fish, which was easier to determine that there was an extra piece. Nowadays, we would send off, the first thing we'd send off is chromosome microarray. And chromosome microarray, this is not at all hard to detect. So as an example of moving forward with technology. This is how we determined that the duplication came from the mother and mattered less coming from the father. So basically, these are markers along the chromosome in this region. You should have 2 different chromosome 15s. And you see that both of the grandparents had what you would see the usual markers. Now, the mother should of the grandchildren should only receive this set of markers or this set of markers from the father, in the same way that coming from the mother's mother, you see this 197, 131 over here. But it was easiest to see in this one where there's 3 distinct markers, each identifying a different piece of this region. And that's what led us to know that instead of getting either the 139 or the 147 from the father, she got both of them. And so now you have this father without autism, mother clearly without autism. Some might say, oh, well, she has it, it must not matter. But the key distinction here is she gets the duplication from her father, but as the mother, now her children are going to receive the duplication from their mother, and that leads to the difference in whether they're affected or not. This has since been replicated, and this is one of the most common rare variations in autism, and also in contrast to some of the others of equal frequency. This one is higher. If you have it, you're more likely to have ASD, meaning about 90%. If you have that, will have ASD. This is the region that actually shows you the microarray results, so it shows you what's duplicated. There is a little bit of variation at the ends, but it's this middle region that makes the biggest difference. And one of the things that we looked at, we actually found it looking at this GABA beta 3 and related anxiety genes, partly because of her anxiety and being interested in this. It's probably this UB3A that's contributing the most to the disorder, because we know from Angelman syndrome that is the gene that drives it. This is the most important maternally, meaning coming from the mother, expressed gene in the region, and there's a lot of emphasis on understanding actually some early work on developing trials to reduce the activity of UB3A as a treatment in this disorder, similar to how there's already trials going on to increase UB3A in Angelman syndrome where there's a deletion. GABA beta 3 has actually, just from the latest analysis that I'll mention later, been confirmed as one of the autism risk genes based on some of the sequencing studies that I will show you. So I'm going to talk a little, I'm going to move to CNVs and SNVs, and I'm going to start with CNVs, or copy number variants. Now, the first thing to think about is those chromosome insertions, duplications or deletions I talked about before are copy number variants. Those are ones that were identified. You could see them on chromosome analysis, but with improved technology, we can see much smaller copy number variants. And I think of these as missing a paragraph or a page of a book. They're typically relevant when they delete or add genes. And and they're usually de novo. If you notice the first case I showed you of the duplication 15, that family's unusual, but the parent of origin means that it was new in the affected grandchildren because they were the first that had the maternally inherited duplication. Okay. So these are de novo autosomal dominant. I had trouble with this whole concept because to me, autosomal dominant was something like Huntington, where you have this large pedigree and 50% of everyone has it. This is an apparent paradox because the parents do not have the variant, only the child. But the idea is that it only takes one single allele to have an effect. And also, if this child of these two unaffected parents without the variant did have offspring, they would have a 50% chance of transmitting that variation. So in that way, you could think of it as how a dominant disorder might start after it being de novo. And most of the things that we're going to talk about in terms of risk, the rare de novo events are going to be these de novo autosomal dominant events. And these de novo autosomal dominant disorders occur when the severity of the disorder is enough for less likelihood of having children. It accounts for a child to have a severe clinical condition with unaffected parents because the mutation is not present in either parent. This continues to be one of great interest. 16p11.2, you actually have to have more refined definition of it. We actually are starting to use more of a nomenclature in the physical nomenclature of base pairs that precisely define where this is. This was from our lab, where you can see the normal chromosome number here, the drop in this region, about 500,000 base pairs of DNA, too small to see under the microscope, as opposed to, let's say, the 15q11, q13, which was right at the threshold of detection with microscopic detection with those gray GM-sustained chromosomes. So once this technology developed, which is either done by SNP array or by chromosome microarray, more typically in the clinic, eventually, and even now, you can technically do this with complete genome sequencing, which is where we're headed. It's really more of a cost issue. So this defined a new syndrome. This is zooming in, very similar to the 15q11, q13 region. We become interested in the genes in the region, sequence them fairly quickly, and no specific gene in this region is sufficient to contribute to phenotype. And many of these copy number variants are probably related to multiple genes within the region, either being deleted in what I'm showing you or duplicated as well. So variable expression becomes very important to think about. And here, the 16p11.2 C and Vs are very important in terms of an overlap with schizophrenia. So deletions lead to autism spectrum disorder, intellectual disability, and obesity. The obesity actually being the most common thing that is present for everyone. Duplications can either lead to ASD or schizophrenia. Interestingly enough, there's a flip side where there's thinness and not obesity. Both are rarely seen in controls, meaning their prevalence is their penetrance. If you have the deletion or duplication, you have an effect of it, but the expression varies. So I mentioned, for example, deletions with ASD. Well, initially we were only studying with the sort of most advanced technology people with ASD, but since then it's become clear that the diagnosis with a deletion can range, or deletion or duplication, taking them all together, can lead to ASD or ID or schizophrenia. But also, there are patients that only have language disorders. I would say at least having a speech and language disorder is sort of the minimum impact of this. ASD only about 25%, which shows if we're only looking for it in autism, we're going to see it only in autism. But when things get broadened out to everyone who's being tested, you learn this. One very interesting thing, and it's a good example of an interaction between the common genetic variants, also an interaction with some of the environment that comes related to SES and IQ. There's an average drop of IQ of 30 from the average of the IQs from the parents. And also, it's interesting in that you see this only 25% with ASD. Well, the test of the parents shows an interaction with we're all on the autism spectrum. Some of us are on the opposite end of clinically diagnosed autism. And the parents that were sort of closer to the affected with autism part of the spectrum were more likely to have a child with ASD with this duplication. The parents that had a very high level of flexibility, very strong in social communication, were least likely to have a child with ASD. And this is a very important paper from Marino De Luca about 10 years ago showing that. And that's not just for 16p11.2 CNVs, it's just the most intensively studied in this way. This is showing that where the deletions, duplications matter. And the key here is, and I would have liked to have been able to show this green part is possibly subtracting. There are some of these variants sufficient to cause autism by themselves. Let's think of the duplication 15q11, q13. There are others that are sort of at a threshold, and it may take other genetic risk factors and non-genetic risk factors, and some of those could push over the threshold, some could lower below the threshold. And there are some, and this would be paternal duplication in 15q11, q13, where, if you see ASD, it's that these other factors are sufficient to put someone over the threshold. Single nucleotide variants. Both parents, for example, might have the protein supercalifragilisticexpialidocious, a de novo missense in one of the child's two copies. Let's say this F is replaced by an X. I don't want to try to pronounce that, but as you see, it would change that. Some of these changes, you could actually see changing letters in here that might not make it that hard to pronounce. Others would. And that's a really good example of why some missense mutations disrupt the protein considerably, and other missense mutations have a benign effect. And then there's de novo nonsense. In this case, instead of replacing the X at that point of a nonsense protein truncating mutation, the protein is only made that far, and the rest of it is not made. These are almost always leading to a loss of function of the protein, particularly if they occur earlier in the gene. So we've learned a lot from single nucleotide variant studies. Again, technology enabled when we were able to do whole exome sequencing. The exome is all of the DNA coding for protein, and the emphasis has been on finding these de novo single nucleotide variants in trios of individuals with ASD and their parents. To have the proband with ASD and their parents allows us to know that a change is de novo. And what we've learned overall is there's a higher burden for loss of function de novo SNVs and smaller enrichment of de novo missense SNVs in brain-expressed genes intolerant of missense mutation. Probably not surprising they're brain-expressed genes. And the intolerance of mutation means some genes will tolerate even loss of function, meaning you can get by with just one functioning copy instead of two functioning copies, so that the evidence comes from the genes that you lose one copy or one copy is dysfunctional. And we use a Nomad browser, which is very helpful for looking for which genes tolerate these sorts of changes. This is the last publication from our Autism Sequencing Consortium group, and shows the various data approaches coming in, and we're basically ranking with very high significance these 72 genes from, in a sense, and many times they have higher levels of significance because of more cases found, more or less. What's really interesting about the coloring here is you see these loss of function or protein truncating variants like the supercalia I showed you, and then you also see evidence for this gene SCN2A, which is a sodium channel gene coming from missense variants, as opposed to SHANK3 here, which has, again, about half or so coming from protein truncating variants, but then a large number coming from deletion of the region, and things that were picked up and are picked up under CNV analysis. So this is 72 genes. This was published 3 years ago. The current analysis that has been presented, and we're working on writing this up with the final analysis, is this number is now about 257 genes at the same strict level of significance. And this is, I have to say, has a lot to do with autism has benefited, as has schizophrenia genetics, from incredible amounts of sharing among investigators, as well as participation by so many families. This is just to show, and you'll see this later in a later talk on schizophrenia. This is showing this similar rare coding variants in 10 genes in schizophrenia from about the same time. And it's these genes above this line here, although what our experience from the autism genetics has been that as we get larger samples, the group in this area largely moves up into that area. And the thing that I mentioned is that there's overlap with these 10 genes. 2 of them overlap with ASD genes, this gene called TRIO, and GRIN2A. Now, I note that GRIN2B is also an ASD gene, and mention that GRIN2A and GRIN2B genes code for NMDA 2A and 2B receptors, and these obviously related to learning, the synaptic plasticity, and there's already been work in this area in terms of developing drug treatments targeting these receptors, and it may be that might have different impact if you were someone that had variation in these genes or not. So I think I mentioned about a 10% increase in these rare de novo variants. I want to sort of see how you get to their exonic de novo CNVs, 5% loss of function de novo SNVs, 10%. This is not limited to the 72 or now 257 gene analysis. Rare autosomal complete loss of function, 3%. Anyway, that all adds up to 20%. And so some will tell you that, well, we can find something in 20%, but fortunately, we participated in a study, the Simon Simplex collection, in which unaffected siblings were found. And if you add all those things up, you see about 10% of the unaffecteds have that. So there's an increase of 10% that's coming from those. And in some cases, like those 72 or 257 now genes I showed you, we're getting a better sense of which of those variants matters. But this is the difference between doing a burden analysis, very important for thinking about research, versus an individual patient that I'm seeing where it's very important to note is this particular variant is that pathogenic for this patient. So I'm really interested in the genetics. I think there's obviously interesting points that pedigree I showed you, or the family that I showed you with the maternal duplication 15q11, q13. That mom wasn't going to have another child unless she knew she could test for the presence. I don't know what she would have done with the knowledge, but she wanted the knowledge, and then went on to have a child that did not have the duplication. So there's a whole area of genetics use. I'll mention a little bit about that later. But to me, it's about how, and I appreciated the introduction so much, because the real question is, how do we develop new medications, both ones that are more effective, because we really don't have anything for autism that treats the core symptoms of restricted repetitive behaviors and social communication problems. So how do we develop them? And some of the things we do have that may treat, let's say, associated irritability or associated other psychiatric disorders to have reduced side effects. So Fragile X is an example where there's a long history of moving from gene to fruit fly to mouse to clinical trials. There have been a lot of improvements in trial design. So far, this hasn't led to a clinical trial that's been positive, although some of this, that doesn't mean we're still not on the right track there. SHANK3 and insulin growth factor is an example where there's current clinical trials involved. And I also note that Rett syndrome, which was in the basically psychiatric manual, is often found in girls with ASD, girls with ASD 3 to 5, particularly when you had to investigate a specific gene as opposed to doing a whole exome and CNV approach or complete genome sequencing. Now has an FDA indication for treatment with an IGF analog trofinetide. And that's, I don't think there's enough discussion. It's true that we would like it to be more effective, so more work needs to go into the area, but to have an FDA indication for a treatment for a developmental disorder that overlaps quite a bit with ASD, I think is important. And I think it may not be as much as where we want to be headed, but it's important to have milestones of success along the way. No doubt 30 years ago, I remember people saying, well, you can't treat neurodevelopmental disorders. And so I think we moved from nihilism to actually a pathway to treatment development. So I mentioned 72 to 257 genes, and, in one way, the 2 to 5 genes we thought we were chasing 30 years ago seems, in some ways, well, then we would have focused on those 2 to 5. And now we've got so many, but the interesting thing is to think about how they cluster. And this has been fascinating. If there's this learning pathway, frankly, I was less aware of. In fact, some of this, these led to a Nobel Prize since I learned about the synapse. But this whole pathway, which I would think of as the learning synapse, and if you and this really came from CNVs, if you deleted any of the proteins in this pathway, you had a risk for ASD or intellectual disability, or in some cases, schizophrenia. and first of all shows how fine-tuned this system has to be. And when I say missing, I mean missing one copy, so dosage significant. It's interesting, we found, I think maybe one of the first, this is also called PSD95, we found one of the first cases of this, or maybe it was just one of the set of genes of PSD95, and it is very similar to Shank, where Shank2 is now implicated in addition to Shank3. So some of these have paralogs, very similar genes, and in some cases, filling in the gaps of a table had to do just really with seeing more patients and having more patients in research studies. This is an example I mentioned before that in many cases what we're looking for is associated disorders. I think this is another practical point of genetics, which has pulling out all of autism spectrum disorder, understanding a specific etiology. This is another one. POGZ is the name of the gene. The syndrome is called White-Sutton. And this is another one where there is a group of people that get together, both people with White-Sutton syndrome and their families, and share interests. In fact, this particular family has been very good about getting the word out about her daughter. She's a woman in her mid-30s with ASD. She lives on her own with support, works on her own with support, both from agencies as well as her family. She also has bipolar mood disorder and OCD, and a lot of people in the audience will know that's a challenge. In terms of our current pharmacological treatments. And then this was about 2019 to 2020. She always had a lot of worries, and she had worries about constipation. And what was striking was the fecal incontinence was not a sort of what you see, I guess, much more commonly in children, but you'll see also in others. She didn't have the typical constipation, sort of leaking incontinence. It seemed to be full. And she did get initial GI workup. I think it was somewhat impacted by her tolerance of the procedure, and I think it was also impacted by it occurring in April of 2020. And she developed acute severe prolapse of an elongated colon requiring emergency surgery. Fortunately, it was fortunate that it was an elongated colon because it was easier to find healthy colon to not as a one-step procedure. And what's interesting is in the pediatric group, as many as 15% in this syndromal form of ASD have surgical emergencies that are similar. They're different in pediatrics, but I think one of the fascinating things is when we had gone the traditional route and requested chromosomes, the insurance rejected it. Actually, they rejected microarray, much less sequencing, which was what was required here. A lot of it had to do with age. Like, is it going to matter? And this is an example where there are a lot of people with White-Sutton syndrome who are older but unidentified because it's mostly being identified with new technology in younger patients, having had the privilege to treat patients for so long. I can now see course, and this is an example of letting people know, well, wait a second, they're not just pediatric surgical emergency risk. And I'll just add that another one of these, because you're going to hear later again about 22 Q11 deletion, also known as velocardiofacial syndrome, is another syndrome that is an ASD schizophrenia overlap with medical consequences. The other one I'll mention in ASD is PTEN mutations, which also are a cancer risk variant. So when they're identified, more cancer screening is indicated. So some ethical implications. I mentioned at least a little bit about the complexity of genetic effects and the 16p11.2 deletion and duplication. Recognizing the variation could be so high. And I think sometimes we may give the impression that it's less certain. We may give the impression that things are more certain or we know more than we do. I think it's important to understand this complexity. Subthreshold traits, let's see if I have a slide for this. Yes, I do. So this is an example. If there were, we used to say 5 genes, but let's say there are hundreds of genes contributing to, for example, restricted interests. Well, that genetic risk may lead to, may be a contributor to autism and so restricted interests that someone has trouble engaging in the social world. But it also could be thought of as the ability to focus intensely. And frankly, many scientific contributions have been made by people that have this trait. Anxiety, I think as psychiatrists, you know that this also means, may mean you avoid certain risks. So this is the point that subthreshold ASD traits are often adaptive for individuals without ASD, and frankly, for people with ASD, they may be strengths for them as well. And finally, the number of common variants contributing to ASD or schizophrenia means that you're never going to, people think that you're somehow going to do eugenics and not have a risk for schizophrenia and ASD, but you couldn't do that. All these variants are there. Many of them were there for a reason. And this idea of pre-existing condition, I think, means we're all somewhat predisposed. And, you know, some have ASD and it may present some challenges. Some of them may have been overcome by some. And so more implications. Genetic etiology does not reduce the need for rehabilitation, education, and non-genetic treatments. And the idea is to empower those with ASD and their families. Common risk variants of ASD are shared partly by all. So I've seen parents shift from the refrigerator mother idea of ASD, and I'm glad I mentioned this to people, and if they're not my age, they don't really know what it is, but it was blaming mothers for ASD, similar to having blamed mothers for schizophrenia. But then people go, oh, it's my genes at fault, and the point is we all have genetic risk, and in one context, that genetic risk may be for ASD, but it may also have benefit. Connection to medical complications and treatments. So I mentioned before identifying patients that may have increased risk for medical complications, and I did mention all 3 of those. And then the other thing I mentioned was developing new treatments through understanding of pathophysiology. To me, that's what it's about. I'm not going to see all those treatments developed, but I have patients young enough that they will, I think, benefit from them. And there's already benefits, like I mentioned, with the trofinetide and Rett syndrome. And I want to thank people with autism and their families who participate in the studies. Just even for our group here in Chicago is a large number. Everyone who cares for themselves or others with ASD, everyone who advocates for services and research in ASD, too many mentors to mention, trainees who have contributed and of whom I'm so proud, other collaborators, NIH, I can't thank enough, the Shaw Foundation, Autism Speaks, Simons Foundation, the Academy of Child and Adolescent Psychiatry, and others for funding. I want to add all these things, particularly those here. I would add schizophrenia to ASD as a clinical head for psychiatry and relative, I can't say enough about the importance of moving forward and improving schizophrenia treatment. And I want to thank all of you for your attention. Thank you.
So thanks so much, Ed. Phenomenal talk and certainly thought-provoking. We'll take a couple minutes for questions and hope that you'll be able to join us for the Q&A. So I'll kick it off, and it's a two-part question. So one is, what would be a takeaway you think for someone who is a clinician and working with patients with either an autism spectrum disorder or schizophrenia that you would take away from your research? And then second of all, what are you excited about in terms of either technology or scientific developments over the next few years?
The first one is, I think for this group, is probably thinking about treating the comorbidity of ASD. And I would say so many patients, I don't know. That's a big one. As far as the genetics, I think to think about, we probably should be thinking more about testing than we do. I think that's a discussion to have with people. I think it's happening at the pediatric level more, but if we're treating adults, we need to think more. I didn't mention, I think I did, the patient that insurance disallowed, the father self-referred, although I refer patients to the SPARK study. And the SPARK study not only is a major contributor to current developments in this research, but also if they find something that's clinically relevant, they actually get back to the clinician and let them know. And either work with the clinician who may, like in my case, feel okay with the genetic counseling, or work with the clinician to also have their genetic counselor work with the family. And the disadvantage, you don't get a report back showing that it's negative, so you're wondering if you're hearing it is research. It's not what I would recommend, but it's a relevant thing to do. And did I miss a question? Oh, what am I excited about?
Yep.
I'm excited about the part of using this information to develop new treatments. And I think we're getting these large sample sizes and just going to the 257 genes. I mean, just like personally excited because now we have these in front of us and we're seeing how they link up with do the ones and genes that are also expressed in the heart as well as the brain. Are they the ones that are contributing to what we need to follow up for the heart? But really at the end of the day, and it may even be treating the comorbidity. I think some of the things that I would like to treat the most is some of this severe comorbid OCD that's often leading to severe problems for the patients and their families.
Super, thank you. Take a look here around the audience and see if there are any questions. Dr. Strauss.
Do you think there's any contribution, Dr. Cook, of mitochondrial DNA variation?
That's a really good question. There's definitely interesting mitochondrial syndromes, and you remind me that I don't think I got an answer to whether our whole genome, I think will pick up mitochondrial DNA. We have to make sure it's not getting masked. I don't think the exomes have been picking up mitochondrial, but I'm not sure. But I really like that question because it's something I asked about, and I forgot whether I got an answer to it. So, excellent question.
All right. Well, thank you so much, Ed. We really appreciate your talk and certainly kicks us off here. So with that, we'll transition to our next speaker. I'm not sure. Yeah, we'll hang on a second. We'll switch slides here. All right. Super. Thank you so much. It's appropriate that we're having technological glitches as we're talking about technology and AI. With that, we'll invite Dr. Rob Marvin to come on up. Rob is going to talk to us about an update on assessment and treatment of psychotic disorders. As all of you know or should know, Rob is our Executive Vice Chair and Vice Chair of Clinical Operations for the Department of Psychiatry and Behavioral Sciences. He's probably most proud of the fact that he's an epic physician builder and a self-admitted nerd. And we're very grateful to have him having joined our department, almost 2 years ago. So lots of exciting stuff that has been happening. So Rob, come on up, please.
Thanks, Ramon. Thanks for everybody who's brought me to this talk and given me the opportunity to give you guys an update on assessment and treatment of psychotic disorders here. I don't have much to declare. I do some lectures for psychiatry board certification exam and past funding from ABPN and NIMH. Today I'm going to talk about some kind of broad-ranging topics. So I'm going to go through very quickly overview, etiology, assessment, and treatment of schizophrenia. I want to shout out a TikTok artist. This is Christopher. This was highlighted in Newsweek a few years ago. He's a filmmaker and has posted a series of short films on TikTok illustrating what it's like to have schizophrenia. And he does a very interesting kind of walk through of what a day in his life would like, or a moment in his life would like, with hearing voices or seeing things or these distractions. And if you've never had one of those experiences where there's some exercises where you can put headphones on and kind of hear what it's like to have auditory hallucinations, he has a really kind of creative and informative way of kind of experiencing what it's like to just kind of have this stuff randomly poking into your consciousness all the time. Okay, let's talk about what is psychosis. Just the way my brain works, I have to have a conceptual model for things. I'm horrible about just memorizing individual facts. That's why I did poorly in psychopharm in medical school. So I couldn't remember all the classes and side effects of individual medications, but give me, this is how it works, I can elaborate on that. So for me, this is helpful in thinking about psychosis. There's a model called aberrant salience, or disruption in the salience network, that's been a model for looking at schizophrenia or psychosis for 20 some years now, probably. And for me, this really highlights kind of the common themes and allows me to kind of think about the different symptoms that people have. So if we think about psychosis, we're thinking about thoughts, experiences that are out of touch with reality. They're not connected to some, like, this is reality, this is not reality. So, okay, that leads us to think, okay, we must have a psychological mechanism in our mind that's making that distinction. This is reality, this is not reality. And that's really the salience network. So there's a network of interconnected circuits in our mind that actually processes internal information and external information to give it relevance or salience. This is important, this is not important. And then our higher cortical areas are putting relevance to that. It's important because it's this. It's important because it reminds me of that. It's not important because I don't need to deal with that anymore. Highly evolved, this is evolutionarily important, because it allows an organism to focus a limited amount of attention on the things that are important. You don't need to pay attention to all these other things going on. You're just going to look at that prey in front of you. There's good evidence now that at least in schizophrenia, there's disruptions in this salience network of how people's minds coordinate the importance of internal and external stimuli. So we think about this aberrant salience as the inappropriate processing of that stimuli that would either be meaningful or not meaningful or irrelevant. And we were able to tie that into some of the dopamine hypotheses of schizophrenia, because dopamine's involved in these different networks. And we can pull that together to account for and represent the symptoms of delusions, hallucinations in psychosis. We're now at the level of data to be able to kind of pull multiple factors together. So looking at different areas of the brain. This little graphic kind of points out different areas that are involved in the salience network. The amygdala, the dorsolateral ventral medial prefrontal cortex, anterior cingulate, hippocampus, striatum, and substantia nigra are all involved. And these have all been previously involved in schizophrenia. The idea around this salience network is that the generation of positive symptoms occurs when stimuli that would normally be irrelevant or unimportant becomes highlighted and tagged as important. And then through conditional learning, the more that's tagged as important, the stronger the connection, the more important that is, we get delusions. Negative symptoms through this network are thought to be the ignoring or discounting, not paying attention to things that people would normally pay attention to in the environment. Things that would be entertaining, or things that I got to go do my laundry, I got to go clean up this thing in the kitchen. Those are not tagged as being relevant or important in the person's internal salience network. And then we have negative symptoms of avolition and amotivation. Okay, so that's my conceptual model, and I can kind of think, no, I can pull all this other stuff into that. Let's take a step back a second and kind of just think about where we have schizophrenia, where it occurs. It's a fairly common psychiatric illness. It's in the top 10 illnesses contributing to global burden under WHO. If we're looking at incidence, kind of the rate of new cases, so the rate of having any psychotic symptom is about 50 in 100,000 people a year. For schizophrenia specific, is about 15. Prevalence is looking at one-time slices, so how many people are currently affected. If we're looking at a 12-month, it's about 4 per 1,000, and lifetime it's about 7.2, which is really where we get like that 1% chance of schizophrenia or prevalence of schizophrenia. The rate of schizophrenia is about even male and female, a little higher in males. Depending on the age of the population, that's skewed more towards males because males have a younger age of onset. So if the population of study is younger, there's going to be a higher incidence, higher ratio of male. If it's an older population, women tend to live longer. It's going to be skewed more towards women. Creates a significant burden. So WHO looks at the disability adjusted life year as a measure. So this is a calculation that looks at lifespan minus, or the disability age adjusted, or adjusted life year is the combination or the sum of the number of years that people have the disability and the years lost to their lifespan. So it's kind of a measure of total disability. And this chart is ranked by different groups in the different columns, or age groups in different columns, and then disorders in the rows. And you can see schizophrenia is ranking pretty highly up there, with the smaller number in each box representing those disability adjusted life years. If you look at some of the other numbers, it's clearly things that are very chronic or very serious illnesses that add to that disability. And then just so we have a frame of reference, looking at the course of illness over time. So if we're thinking about patients with schizophrenia, it's usually a relapsing and recurring illness. So for most people, the premorbid symptomatology is very little. They have very few symptoms premorbid. In their, as they're beginning to have symptoms, they maybe beginning to have some odd pseudopsychotic things. And then most people will have an acute psychotic break where there's a very florid onset of symptoms and acute illness. For most people, they'll have a pattern of fluctuating symptoms over their 20s and into 30s. For most patients, by the time they get to their 40s and on, they have more stability and less severe symptoms for most patients, less hospitalizations. Okay, what do we know about what's going on? Etiology. The current model is what we would call a diathesis stress model. So this is probably the same model we have for all of our psychiatric illnesses. So it's a combination of the individual's pre-existing vulnerability, so the diathesis, and then either stressful life events or environmental factors contributing. Said another way, neurodevelopmental substrate disruption that interacts with the environment and social stressors that produce the syndrome. And we have evidence from that from various lines of research. So neurotransmitter data, anatomical data, genetic data that we heard like a great talk from Dr. Cook on, and then certainly environmental data as well. Let's talk about neurotransmitters first. Dopamine is the most researched neurotransmitter probably in schizophrenia, so there's very, very strong evidence that dopamine dysregulation is a primary end result or cause of schizophrenia. We have strong evidence from that because our medications, almost all the medications we have, are dopamine D2 receptor antagonists. And then in the inverse, substances or drugs that increase dopamine cause psychosis. So amphetamines and PCP act in that way. With dopamine, we have fairly well delineated pathways in the brain that can interconnect different parts of the brain, and we can roughly correlate the dysfunction in those pathways to specific symptoms, like positive symptoms, negative symptoms, and side effects. We also have good evidence from serotonin. Primarily, the evidence from serotonin is really our medications as well. So our second-generation antipsychotics are very potent in serotonergic activity. And then we have evidence from substances as well, like the psychedelics that cause psychosis are serotonergic medications. So it's either antagonism, like blocking serotonin receptors, or inverse agonism, attaching and decreasing the activity at the serotonergic receptors that potentiates the action of the medications. And the thought that from serotonin point of view is this is a regulatory or co-regulatory mechanism for dopamine metabolism or dopamine release. So you can regulate dopamine release by regulating and improving serotonergic function. And then we also have evidence from the regulatory neurotransmitters in the brain. So GABA is an inhibitory neurotransmitter. It's kind of the brakes of the brain. There's evidence that loss of GABAergic neurons unleashes or takes away that regulatory function and then increases dopamine and causes psychosis. Glutamate is a activating neurotransmitter. Also is involved in regulating neurotransmitter dopamine release. And this is how PCP and ketamine potentiate psychosis as well. We have good evidence now from some of the genome-wide association studies that these GABAergic and glutaminergic receptors are involved in the pathways for schizophrenia and psychosis. So this slide is kind of a summary of what we've got going on with the neurotransmitters and the actions of this. The panel on the left-hand side is kind of the areas of the brain that are affected, the frontal cortex that are in blue, striatum in red, and temporal cortex in green, showing the differences in neurotransmitter activity in the different sections of the brain. And then the panel on the right is the linkages. So the idea is that in this pathway, the lower cortical glutamate affects the GABAergic neurons, affects GABA levels, inhibits the cortical control of dopaminergic neurons in the striatum, increases dopamine release, relating in causing those psychotic symptoms. Okay, let's look at anatomy here. This is a long-standing feature or a pattern that we've seen in patients with schizophrenia is loss of brain volume over time. When we just had x-rays or CT scans and then onto MRI, we've evolved our level of detail from kind of gross anatomical brain atrophy or decrease in volume to very specific areas of the brain that we can now look at with high resolution MRI. And if you look at these are identical twins, kind of a well twin and an affected twin. You can see the lateral ventricle size. So the little butterfly lateral ventricles are much larger in the affected twin. On the bottom panel, this is much more dramatic. And you can also see widened sulci across the top here, representing brain atrophy. So that's kind of a gross, less brain volume, less brain cells type of thing. If we look at kind of studies that have looked at more precise measurements of brain volumes, it's in those areas that we've implicated in the neurotransmitter dysfunctions. So the amygdala, the caudate, frontal lobe, hippocampus. And then also on this slide is the ventricles. Those bottom two rows, the third ventricle and the lateral ventricle, are greatly increased in size in patients who are affected with schizophrenia or psychosis. So loss of brain volume, loss of brain function. So why is that? The ultimate answer is we probably don't know. One of the theories out there is that there's some aberrant synaptic pruning. People remember this theory? Yeah? So synaptic pruning is why we can't learn Spanish at 30. Our brain undergoes a process of refinement. The idea behind it being that we're going to eliminate, decouple those connections that are weak and enhance those connections that are strong so that we can remember what we've learned growing up and survive. Okay? Errors in this synaptic pruning are thought to be one of the reasons for the onset and timing of schizophrenia. So something about the synaptic pruning process sets off the symptomatology or unmasks the vulnerability, maybe. Kind of connecting it to autism, the idea around autism around synaptic pruning is that there is insufficient synaptic pruning and there's too many connections left. And so that's the dysfunction in autism spectrum, versus schizophrenia where there's an over pruning and decrease in brain volumes. Some other evidence we have, so looking at this from a different way, functional imaging. So this is looking at a functional MRI. So putting someone in an MRI machine, giving them a specific cognitive task, and then looking how the different parts of their brain are activated when they're actually doing a cognitive task. It is good at looking at very specific cognitive functions. So it's not necessarily a global picture, but it's more of a individual thing. So looking at memory, social cognition, and really looking at that salience network as well. Another way to use the MRI machine is just put someone in there and let them just be at rest. And so what is someone's brain doing when you're just kind of lying there thinking or just your mind is wandering? And they've showed some differences between controls in patients with schizophrenia and just how their brain idles. Again, connected to the salience network and kind of the cognitive functions that are running in the background. Okay, let's turn to genes here. We heard a lot from Dr. Cook about different genes involved in autism spectrum, and we're going to kind of talk a little bit about that here. So starting with just inheritable risk. So if we start at the bottom and look at monozygotic twins, there's a 50% chance of inheritability. So if one twin has schizophrenia, 50% chance the other twin might get it or present with that. As we go up the chart, the degree of relativity decreases, and so it rapidly decreases. So by the time we get up to like second level, like cousins, aunts, and uncles, the risk is just slightly above the general population. This is kind of a callback to Dr. Cook. So we've looked at tens of genes, hundreds of genes maybe now in schizophrenia. And they are falling into those similar buckets that we have with autism. So they're copy number variants, they're deletions, they're de novo gene mutations. And then looking about where all those genes are working at the cellular level, all those little purple ovals there are different genes that are associated with schizophrenia in these studies, and where they're acting within the synapse and nucleus of the cells. So we've got a lot of data that there's a lot of different genes that contribute to the risk for schizophrenia. So where does that come together? So I like this slide because it kind of pulls a lot of the genetic data together, or the variance in the risk for schizophrenia. So if you look at that inner ring, the green ring, that's the just general inheritable risk, the 20 20% that's non-inheritable, and then the 80% that's just heritable in total of the risk. And then of that 80%, we know, I don't know, what is that, 25, 26, 7, 8, 29% of it, we know where it's coming from on the right or the left-hand side. And then of that piece, we kind of know a little bit more about where some of those genes are coming from. So there's big chunks of genetic inheritability that we don't know exactly where it's coming from, but we've come so far in 20 years to identify these genes. Okay, let's look at some environmental factors. So this was a thing that was probably more common when I was in medical school and in residency, was maybe looking at these risk factors, like the importance of different environmental factors in schizophrenia. So these are all like a summary of different things that incur at least some risk for development of schizophrenia. So obstetric complications. So if a person is born at the time of their birth, there's obstetric complications, that incurs some risk. Born in winter in the northern hemisphere. Childhood trauma, urban living, being a first-generation immigrant, and then cannabis use. We'll talk about that in a second. The thing that I kind of attach all these things together, these are all neurodevelopmental risks. So a kid who has a developing brain, their brain is sensitive, their brain is malleable, their brain is going to be influenced by environment and stress. And all of these things are representing a stress to a developing child's brain and increasing that risk of schizophrenia in the context of maybe having some set of genes that puts them at risk to begin with. Okay, let's talk about cannabis. Cannabis and psychosis do not go well together. Great evidence that says the more you smoke, the more you're at risk. The panel on the left is a risk of developing psychosis based on exposure. So the more you smoke, the more at risk you are. And those are just the different lines of the different studies that looked at that. And then the panel on the right is kind of combined or summarized results. So the risk of developing either a psychotic disorder or having a diagnosis of, frank diagnosis of psychosis or schizophrenia from risk from using cannabis. So bottom line is, if people are at risk, they really shouldn't be smoking. I mean, there are hundreds, thousands, millions of people out there that smoke all the time and never become psychotic. So we know that that's not a one-to-one relationship. But people who are at risk, strong family histories, clear cause and effect relationships for patients, really should be addressing that. Okay, so let's kind of pull all this stuff together. So we know there's some nature. This is the genetics. This is the developmental processes that are coming together, the role of neurotransmitters, maybe stress vulnerability, combined with nurture or the environment, what developmental experiences, life stressors happen for the patients, their exposure to cannabis. This leads to multiple areas of brain dysfunction. So we have problems with connectivity, neurotransmitter dysregulation, that synaptic plasticity, pruning, synaptic strength. This is that idea of that salience network and how those things are coded together. All of that resulting in our symptoms that we see. So the hallucinations, delusions, thought disorder, affect changes, cognition, and we call that schizophrenia. Okay, so we know where it comes from. How are we diagnosing this? Okay. Some things to remember. These are the diagnostic pearls here. So there's really no specific sign and symptom that's just going to say, okay, this person has schizophrenia. So it's a process. We know that the duration of untreated psychosis results in worse outcomes. So when you're seeing a patient, if you have experience, you can probably guesstimate how long they've been psychotic by the severity of their symptoms, how well developed their delusions are. So you can kind of just guesstimate some of that, I think. Some things to remember, that the symptoms are going to vary over time, especially early in the course of schizophrenia. They're going to be waxing and waning. Patients will have days, weeks of symptoms, and then resolution of symptoms, and then 6 months later might have some more symptoms. So it really needs to be a process. We need to take developmental factors. So is this person 14 when they're having these symptoms, or are they 24? A person's life events and where they're at, how much education they've had, really varies between that 10 years, and how they present and their understanding of the illness is going to be very different. In the end, our goal is really the same, though, really trying to identify target symptoms, have some measurable outcomes, and promote care and recovery. Okay, I talked a little bit about this, but when we're thinking pre-morbid, for most patients, you wouldn't notice much going on. Sometimes you can identify those risk factors like a stressful event, some abuse history, but again, those are kind of retrospective things you might know, because there's a lot of people that have had those same environmental exposures and haven't had schizophrenia or symptoms at all. The prodromal phase, so this is the period between first onset of symptoms to kind of frank episode of psychosis, really varies between people as well. This can be anywhere from days, weeks, months to years. During this pre-morbid prodromal phase, people can have some pseudopsychotic things, some intermittent kind of frank psychosis that in the slice of time might actually meet diagnosis for schizophrenia with resolution of that. Important to think about comorbidities. So anxiety is very, very common. If you think about that, just having people starting talking to you or starting to see people on the street that are starting to follow you, that's going to be anxiety provoking. So those are often the common things that people might experience and actually might bring them in. And then that'll affect different things, behavior, sleep, activity level, that thing. And then we usually see decline in functioning as a result. So difficulty going to school, staying at work, that type of thing. So assessing. So we want to do a good clinical interview, obviously, psychiatric, substance use, medical histories. For these patients, getting collateral is almost essential. For people in their first episode, their experience, they're going to be naive to what psychosis is for the most part, or what schizophrenia is. So for them, that experience is real, and these are things that are truly happening to them. So you often need collateral to be able to confirm onset and impact in life and that type of thing. There are different rating scales that have been developed for assessment of psychotic symptoms, so you can track over time. The DSM-5 actually has like what they call the cross-cutting symptom measures now, which are basically tracking for symptom domains rather than specific diseases. Certainly assessing for risk is important. We'll talk about that in a minute. And then if we're doing antipsychotics, we want to make sure we do baseline aims. Okay, symptoms. This is just a busy slide with all the symptoms. So these are things we're going to look at. Patients with schizophrenia or psychosis often have disorder in the formation of their thoughts. So this is what's called a formal thought disorder. So being able to put one's thoughts together. They have a disorder in the content of their thoughts. So this is delusions that we'll see, or ideas of reference, thoughts that are inserted. Usually we talk about the delusions as having a particular theme. Hallucinations are sensory information. So this is auditory hallucinations, visual hallucinations. Auditory is much more common in schizophrenia than visual. If a patient has hallucinations in another sensory domain, start thinking maybe about something else. So epilepsy would be a common thing where patients have other hallucinatory events. Substance use or withdrawal would be another place. Illusions are the misrepresentation or misperception of true sensory data. So this is the seeing someone's shadow or seeing shadows behind them and maybe thinking someone's in the shadow type of thing. The shadow's really there, but people are misinterpreting the intent of it. As we talked before, behavior can be really significantly altered, as well as people's affect. So they can range from kind of a blunted affect, restricted affect, to really labile, variable. And then we know from neurocognitive studies that cognition, so concentration, abstraction, memory, executive control are all affected. Suicide. We need to talk about risk for patients with schizophrenia for sure. So it's the leading cause of premature death in schizophrenia, suicide. Between 25 and 50% of patients will attempt, and between 5 and 10% will succeed. So it's very high risk. It can really occur at any stage of the illness. And as a reminder, kind of the risk factors for general population, male, white, single, socially isolated, unemployed, positive family history, previous attempts, maybe some substance use, depression in there, stressors. So this is kind of that classic person who presents to the ED, newly divorced, losing their job, started using alcohol or substances again. That's kind of that classic thing that we're kind of taught, I think. When we're thinking about schizophrenia, especially first episode schizophrenia, there's some other factors that kind of pull it up out of that or make things a little different. Higher level of education, greater insight, low negative symptoms, high level of mental suffering, agitation, presence of insight. So this is, we kind of talked about the suicide crisis syndrome, and this is kind of that overlay into schizophrenia. So this is the young patient who has enough insight to recognize, hey, I might have schizophrenia. My relation to schizophrenia is that person I see on the corner who's talking to himself. I don't want to be that person. I can't deal with this. This is really rough. So that's what makes those kind of different than kind of the general population. Things we really need to watch out for. Okay, so let's think about how this plays out. So for most patients, they're going to be what I call on like the schizophrenia pathway. So they may present originally with like brief psychotic symptoms. We have at least some months or days of syndrome or symptoms. Now they're schizophreniform. They now have this more than 6 months. Now they have schizophrenia. So a lot of people will be on that pathway. And then the other psychotic disorders include schizoaffective disorder and delusional disorder, due to medical or substances, and then the other categories. I'm not going to go through all of these. These are just here for your purview. So these are just the DSM for brief psychotic disorder. Couple highlights. Brief psychotic disorder doesn't have negative symptoms, so they don't need to have negative symptoms to get brief psychotic disorder. Schizophreniform disorder is really the same as schizophrenia, except timing. So it's 1 month to 6 months. And then schizophrenia diagnosis is 6 months or more. As with a lot of things in DSM, you can multi-code things, where they're at in their episodes and if they have catatonia or not. A couple DSM-5 highlights in schizophrenia. They removed the requirement that delusions be bizarre. Because I think just over time, or maybe just with technology, the determination of what's bizarre enough or bizarre became harder to determine, I think. 50 years ago, if someone thought, okay, I have a chip in my head that's recording everything that I see and do, 50 years ago, that would be bizarre. Today, I'm carrying a device around that's tracking my every movement. So that was one of the reasons they took that out of there, I think, because it wasn't really helpful, and didn't predict outcomes. If they had or didn't have bizarre delusions, did they do better or worse with treatment? No. Same thing with the Schneiderian first rank, like were there two or more voices commenting or not, was the content of those. And then similarly with schizophrenia subtypes. So were they paranoid, were they disorganized? Got rid of those because it didn't really impact treatment and outcomes or be predictive of outcomes. And just clinically, people could kind of go between different types. People may present with disorganized and then they come down a little bit and now they look like paranoid, and it's really hard to keep track of. Okay, little timeline. So brief psychotic disorder, remember, is just a month. Schizophreniform disorder is 6 months, and then schizophrenia is at least 6 months. And this includes prodrome, active, and residual. So if people are in treatment, but they have some residual symptoms, you got to include that in your timing. Schizoaffective disorder, it's highly overdiagnosed, I think, but it's hard to diagnose, I'll say it. So you have to have a mood disorder. So you have to have either depression or bipolar, like mania and depression. And then separately, you also have to have a psychotic disorder. So the differentiation between like a mood disorder with psychotic features is that you have to have at least 2 weeks of psychotic symptoms in the absence of mood symptoms. which is kind of hard to determine if someone presents to the emergency room or the inpatient unit and they're highly depressed but highly psychotic, or manic and psychotic. It's hard to pick out. You really have to follow them longitudinally to say, okay, their depression is better, their mania is better, but they're still psychotic. So this little graph kind of just outlines some of that stuff. So to be schizoaffective disorder, you really have to have that pink hump in the beginning where they've had frank psychotic episode without any mood symptoms in order to get there. Catatonia. Catatonia is probably under-recognized, I think, a lot of times. Catatonia is a psychomotor dysregulation syndrome, and it has lots of causes. So it can be caused by a primary psychiatric disorder. It can be caused by medical disorder like metabolic disorders, sepsis, that type of thing. End result is people are really sick, and they really need a high level of care usually. So in DSM, we can categorize it connected to those things. So is this part of a mental condition? Is it part of a medical condition? Or we don't know, really. I'll put a shout out to the University of Rochester. If you're interested in catatonia and you work with patients that might have catatonia, go look at the University of Rochester Department of Psychiatry catatonia website. They have videos on how to actually do the Bush-Francis scale. They have demonstration patients where they're doing the exam on and then talking through the exam and kind of why they rated something this way or that way. It's a really good informational site for catatonia. Catatonia has these motor symptoms. I'm not going to go through this because I'm going to run out of time here. Substance or medication-induced psychosis. So this is where we have direct evidence that the psychotic symptoms are due to either the medication or the substance, and there is a physiologic connection between either the substance and the medication to cause these things, either in intoxication with the medication or withdrawal from the medication. Medical causes of psychosis. Delirium until proven otherwise is my kind of rule of thumb. So delirium is probably the most common reason while patients are in the hospital or they might be experiencing psychotic symptoms, like hearing things that aren't really there, seeing things that aren't really there, maybe being a little paranoid that something's going on. And delirium is really a medical illness. So there's some medical condition that's causing brain dysfunction that's resulting in psychotic symptoms. But there are other medical conditions, neurologic conditions that can contribute to psychosis as well. Lots of different substances. So here's the list. I'm not going to go through those. Okay, let's talk a little about treatment. So our treatment plan. Given our etiology, we have to provide treatment that's kind of going to be matching these different risk factors and arms of what we know about what's going on. So we really need to make sure we've got the accurate diagnosis first. What's going on? Is there something treatable? Is this a medical thing? Is this a substance we can get rid of? Put a treatment plan together, working on a therapeutic alliance with both the patient, the family, educate them. When I did my first episode work at UIC, this was probably our primary goal, was educating patients and families on what is psychosis, what is schizophrenia, how do you manage when your son or daughter is starting to have more symptoms. Just giving people that information and have them be active managers in their illness can go a tremendously long way to preventing downstream effects, I think. So the more we can educate and do that upfront, the better outcomes we can. Obviously, treating comorbid conditions, attending to social functioning is really important as well. So getting people back to school, work, whatever their goals are. And then like that cross-continuum handoff documentation. So almost all these patients are going from inpatient to partial to outpatient to inpatient to partial, back and forth, and having some continuity across that system and that those service lines is really important. Okay, let's talk about meds. Meds are our primary treatment to decrease the symptomatology that people are having. We've had meds since the 50s. Thorazine was our first marketed medication. All the medications that we have, except for a couple, now on the market, new medications on the market, all work by regulating dopamine in some way. We have the two medications now, that Nuplazid and the xanomeline, work by a different mechanism. One just on serotonin, and the other is a kind of a new mechanism action looking at muscarinic cholinergic receptors. Our medications have tended to be divided into two categories. So the first generation antipsychotics, they used to be called the typical antipsychotics, much more directly active on dopamine. And then the second generation medications are more broad spectrum, I guess. So they work more on serotonin or some other combination of medications. I'm a fan of old advertisements. So these are some of the marketing for Thorazine, the one that came out. The one on the left, when the patient lashes out against, quote, them, Thorazine quickly puts an end to his violent outburst. So a little Twilight Zone action there. For prompt control of senile agitation, use Thorazine. Thorazine can control the agitated, belligerent, senile, and help patients have a composed and useful life. What's a little judgy there? And then there's one for prompt and sustained relief from severe mental and emotional stress. So, housewife upset about something. Hopefully you've come away from that. So how do we choose medications? We've kind of moved away from strict medication algorithms. So if you remember years ago, like I grew up in TMAP, which was the Texas Medication Algorithm Project, which was this very algorithmic decision tree about use this, then this. If that doesn't work, use that, then that. It provides general good guidance, probably. The theming is okay, but because we have more medications now, we kind of know more about how medications work and they don't work. Having those strict algorithms in today's age really doesn't make as much sense. It may not be as clinically useful. We know now that with our medications, there's a lot of heterogeneity between how they work. And we don't have a lot of evidence on efficacy. Like one is, aside from clozapine, a medication that's going to be much more efficacious than another medication. So the actual choice of it depends on a lot more factors. Again, with the side effects, we don't have a great way of predicting which patient is going to have more side effects or less side effects, with the caveat now that we've got some of that pharmacogenomic metabolic metabolism data to kind of guide us in that. Okay, so how do we do it? We have to take all of this stuff into account. So we have to look at the efficacy and the side effects and what the patient's preferences are, what's important to them as far as a side effect or a side effect. Do they need something for sleep? Do they want to avoid something that's sedation? What type of formulation do we need? Is this a patient with difficulty with adherence, so we need a long-acting versus not? Are they taking any other medications, so we need to worry about drug-drug interactions? If we're doing multiple medications, are we looking at the receptor profile to make sure we're not duplicating a mechanism of action that we've already got on board? And then those pharmacokinetic and pharmacogenomic considerations as well. So this would be like the rough outline here. So choose a medication based on those factors. First generation, second generation, clozapine or long-acting, depending on the patient population and where you're at. If they have a good response, yeah, keep going, they're good to go. If it's a side effect issue, probably go back and try another medication in those first generation, second generation one, because we're just trying to look at, it's not an efficacy issue, it's a tolerability issue. So we can probably choose a lot of different medications. If it's a efficacy issue, most treatment algorithms will start saying, well, consider using clozapine early. If you have a good response, move them down into maintenance. Now, if they failed a couple of medications, if it's tolerability again, pick another medication because it's not an efficacy thing. If it's now efficacy and they failed two medications, really start thinking about clozapine. There's good evidence to say that if people have failed at least two antipsychotics, and they're good trials, good doses, good timing, you can ensure the adherence, they're not missing doses. If they fail those two, there's good evidence they will continue to fail. So we got to do something different at that point. So just trying another single agent probably is not good. So think about clozapine, think about maybe doing some combination at that point. And then if that's not working, ECT. So ECT has good evidence in acute treatment for schizophrenia, not good maintenance data. So you have to use antipsychotics along with it. You can't just use ECT. But in the acute course, if someone has failed clozapine, ECT would be the choice. And before we had medications, that was the treatment of choice for schizophrenia, was ECT. Bringing back the neurotransmitters. So this is a diagram of the neuron and then where our different medications are acting. So we've got the dopamine here at the receptor. So we've got too much dopamine going on. Dopamine antagonism is blocking these receptors and having its effect here. Serotonergic medications are working higher up. So they're working at the regulatory glutamatergic neurons and controlling those. Muscarinic activity is up here. So we have different kind of methods of action now. And if we're thinking about combining medications, that's what we should be doing. So if we're have a medication on board that's highly dopaminergic already, let's look at something that's actually maybe working through serotonin if we're going to combine things together. Okay, very busy slide, but efficacy data. So we have very good what they call network analysis of medications. So they take thousands of drug studies with thousands of patients and do a network analysis to look at comparing different components of the medications or like efficacy and then side effects. So on the far left is overall change in symptoms with the different medications. So it's probably too small for anybody to read. Clozapine is up at the top, and it's up at the top by a fairly significant margin to the left. This is both American and European medications, so some of these medications we don't have. But if you look at them, you can probably draw a line. Like the bars are the 95% confidence interval, so we're 95% sure that the true value for that medication is in that bar someplace. We can probably draw a line that goes through all of these, like from top to bottom, almost all of them with a single line, meaning that there's not going to be much efficacy difference between them. And then it's broken down by positive and negative symptoms too. So this middle column is positive symptoms, right-hand column is negative symptoms. Again, clozapine is at the top for the charts for both of them. Other things at the top that we have here, olanzapine, risperidone, thorazine, paliperidone. So one way to think about medications, if you're just kind of saying, okay, I want to go for most efficacious, okay, let's look at this. We can at least go down the list that way. The other way of looking at it is side effects. So if we're looking at side effects we want to either use to our advantage or avoid, we can look at these medications from that point of view. So on the far left is weight gain, kind of in the reverse. So the things at the bottom are the medications that cause the most weight gain or favor placebo, and the things at the top of the chart are the ones that are the most weight neutral. So ziprasidone, people lost weight compared to the placebo in that case. Down at the bottom, olanzapine, clozapine, quetiapine, the ones we think about are down there. And then also QT prolongation and sedation are the other two charts. So this can kind of guide us in making a selection or thinking about our next step. Shout out to clozapine. It's our most effective antipsychotic. I'm a fan of clozapine. We underutilize it because we think it's more complicated than it really is. It's definitely has high efficacy and does incur some more risk. So we have to think about that and manage that. But great data to show people will respond. So in patients who have failed 2 or more trials, the response rate says roughly 2/3. So 2/3 of people, or 1/3 of people will have a dramatic or substantial improvement. 1/3 will have a modest improvement, and about 1/3 will have a mild or no improvement. So in a person who's failed multiple antipsychotics, if we can get 2/3 of them to have at least a mild or moderate improvement, that's a win. It's also shown to dramatically decrease suicidal ideation in patients with schizophrenia and schizoaffective disorder. So if that's a component of someone's illness, clozapine is a good option for that as well. If we think about just kind of a very simple risk-benefit ratio, the risk of death from clozapine is about 2 per every 10,000 patients. If we compare just that compared to like the rate of suicide, and we kind of put it in the same numbers, it's like 900 to 1,300 per 10,000. So if we can make a decrease in that risk, that's a good use of clozapine. Medically, the biggest risks are agranulocytosis, so decrease in neutrophils, myocarditis, cardiomyopathy, and pulmonary embolism or DVT. Those are the acute medical risks that we're most concerned about. Most of these have happened within the first 6 months of treatment. Most of them are the myocarditis, cardiac, and the pulmonary embolism people are really sick. The ANC is not something that you would notice until people are pretty affected. So when clozapine came to market, there was a requirement that we monitor the ANC. Well, not acutely. Aftermarket, when it came out, we noticed this was a problem, started the REMS study. So the requirement was that we'd had to monitor absolute neutrophil count weekly for the first 6 months, every 2 weeks for the next 6 months, and then every 4 weeks or monthly after that first year. In February of 2025, they just changed the REMS requirement so that we have to continue to do the blood monitoring on the same frequency and assessing that, but we don't need to send it into the central REMS program in order for it to be dispensed anymore. So a little documentation burden relief there. Another thing we need to keep track of is metabolic syndrome. So this is common with second-generation antipsychotics, problems with increased weight, triglycerides, blood pressure, increased glucose. Again, we have data on medications that are more at risk or less at risk. So if we're thinking through these things and we know that someone's at risk, we should maybe pick a medication that's a little more neutral. Some acute side effects that we pay attention to are related to dopamine. So the first 3 columns, akathisia, dystonia, and Parkinsonism, are the result of excessive dopamine blockade at the synapse level. Different treatments for those, mildly, moderately effective maybe. Usually if someone has one of those side effects, we'll try and switch medications. Tardive dyskinesia is one of the chronic symptoms of excessive dopamine blockade. Has different risk factors associated with it. We have a couple new medications that valbenazine and dutratebetazine that we can use now. Put this as a slide in there just for more information about tardive dyskinesia and some of its assessments there. One of the questions is, what risk is there for tardive dyskinesia compared to medications? Not a lot of great comparative data. So even looking at this table, this isn't really a comparative risk table. This is a risk of TD in the studies for these different medications. So it depended on the population and the patients studied, but it gives you at least some idea of kind of the risk, with the second generations having much lower risk than the first generations in general. So a big thing, we have to screen for it and monitor for it over time. As I said, we have a few treatment options now. Okay, lastly, I wanted to give some information on some psychosocial interventions that have some good evidence or great evidence for helping patients with schizophrenia. This summary slide lists the ones that have at least decent level of evidence and how they've been helpful or how they've been studied. The challenge we have is how we should best combine them. So this type of person with this type of needs should get this type of intervention. So that's kind of yet to be determined and really up to clinical things. One area that's shown really good evidence is the coordinated specialty care programs. So this is like a first episode psychosis program where you have intensive, kind of like a model on the ACT team or something that's really dedicated towards first episode psychosis. So this was a 2-year first episode psychosis program. The panel on the left is looking at readmission rates. So the readmission rates, the green line is the program participants and the orange line is the non-participants. So the risk of readmission is less over time. And then the right panel is all-cause mortality in users. So really showed a dramatic stabilization of patients' health and mortality over time in those 2 years. I talked a little bit about assertive community treatment. So this is a really an intensive outreach, community-based model. Patients need to be screened for this. So the patients that do wellness program are high utilizers, so lots of admissions, chronic illness. It involves intensive case management, helping patients stay in the community, and it has that good effect on housing, employment, and core illness. And then also CBT. This is kind of a niche area of CBT, so CBTP or CBT for psychosis. It's a modification of kind of the standard model of CBT. Looks at modifying the delusional hierarchy. So a delusion is a fixed false belief, which is very similar to a phobia. If I have a phobia of elevators being dangerous because of my past experience, I can use CBT to challenge that. So if someone has a fixed false belief that white vans are following them, and they're not actively psychotic, you can use CBT kind of challenge that and help patients kind of conceptualize that was part of their illness, and that isn't really a true thought. That's an irrational thought now based on their illness. Great data for this to show effect over time. So if you do an intervention with someone, they have like 5 and 10 year studies to show that this is the lasting effect for patients. They can learn to manage their symptoms and learn about their symptoms better. Okay, so we looked at nature, where things come from, risk factors for nurture, environmental exposures will contributing to this brain dysfunction, symptom production, and resulting in our diagnosis of schizophrenia. We have good evidence now on where this is coming from and really informing our treatment models now. I didn't have a chance to add this, but we've got some new targets for medications and kind of some emerging therapies like TMS, looking at very specific regions of the brain. I just want to thank everybody and thank all the patients I've worked with, colleagues I've worked with. Thanks.
Rob, why don't you stay up here? We'll give an opportunity in case there's 1 or 2 questions before we break. John.
I'm just curious about the new change in REMS, getting rid of that clozapine, and how you see that clinically changing your clinical practice when it comes to surveillance labs, et cetera.
Shouldn't change anything clinically because the prescribing instructions are the same. So we have to monitor everybody at the same frequency and keep track of that. We just don't have to report it to REMS anymore.
I guess my other question is, Europeans kind of monitor it differently, so I'm curious if you have thoughts on that philosophically.
No. A different way of doing things, I think, yeah.
It's a little looser, less frequent, and I'm curious that when the risk, that surveillance itself sometimes keeps people from prescribing it in the first place.
For sure, and I think that's part of the reason they made the change was to improve access, I think. It's probably, it will evolve over time, I hope, because at each one of the committee meetings where they've gone through this, those things have been raised, and so this is probably step 1 of a multi-step transition, I would say.
So I'll ask a quick question. You didn't talk about long-acting injectables too much, so if you could comment on LAIs.
Yeah, sure. Also a big fan of LAIs. So long-acting injectable medications are a formulation of one of our regular medications that have a long half-life, and they've got a lot of technology in them, these little microspheres and the ways of kind of slowly releasing them so that patients will get an intramuscular injection either in the shoulder or maybe the hip, and lasting from anywhere from 2 weeks to 6 months now. We have one. There's also some new sub-Q injections. Rossetti is a sub-Q one. They are quite helpful for patients if they've had non-adherence. So I guess my rule of thumb, I have a very low threshold. So if someone comes back from a hospital discharge and they've had kind of any difficulty in maintaining adherence, they should be asked about, do you want to just take an injection so that can help you just keep up with your medications? Great evidence to show that keeping on a long-acting injection allows people to get into the recovery pathway. So we need them to have their symptoms under control so that they can get back to doing what they were doing before they became sick. So that is really our goal with that. So get them adherent or get them on something that maintains that medication.
Super. All right. Thank you, everyone. Very much enjoyed that presentation, Rob. Thanks. So we're running a smidge behind schedule. So we'll take a 15-minute break. So I've got 10:08, so we'll plan on being back here at 10:23, please. For those of you online, plan to be back online about 10:23. Okay, thanks, everyone. We're going to go ahead and get started. Thank you all for coming back fairly promptly. Appreciate that. Hope that folks are online. A great first half of our symposium, and now we're in the home stretch here. So I am pleased to introduce our next speaker, Dr. Jubao Duan. Jubao is the Charles Walgreen Research Chair and Professor at the Department of Psychiatry and Behavioral Neurosciences at U Chicago, and Director of Stem Cell and Functional Genomics in Psychiatry, as well as the co-director of our Center for Psychiatric Genetics at the Endeavor Health North Shore Hospitals in our Department of Psychiatry and Behavioral Sciences. I was just telling Joao when I first met with him a couple years ago, one of the things that struck me in that meeting were two things. One, that I probably didn't understand 95% of what Joao was explaining in terms of his research, so hopefully we'll get a clearer sense of that today, because it's really brilliant and amazing. And that secondarily, if there was anyone in our department who's going to win the Nobel Prize, it's Jubao. So definitely very excited to welcome Jubao to talk to us about psychiatric genetics and functional genomics in schizophrenia.
Okay, all right. It's a great pleasure to be here. Thank Roman for a kind of introduction, giving me a lot of pressure. I have to let me press it. Anyhow, my topic here today would be more like, okay. All right, so, it's really honor to be on the same stage as Dr. Cook, who spoke earlier. As I mentioned to Roma, when I came here as a postdoc more than 20 years ago at the University of Chicago, Dr. Cook was a professor there. I learned a lot of genetics from Dr. Cook, and also Dr. Alan Sanders who sit there, and also Dr. Gedgeman. We introduce in next slide. So today, I'm really honored to on the same stage as Dr. Cook, and also great pleasure to be here to share my research. So before I go more in detail in science, I want to give credit to Dr. Pablo Gedgeman and Dr. Alan Sanders. Dr. Gedgeman established the center in 2003, more than 20 years ago, when we first moved here at North Shore. He retired a few years ago, and since then, Dr. Alan Sanders and I co-direct the center. So initial research focus of the center was to identify, genes that contribute to schizophrenia risk and other neuropsychiatric disorders using linkage and also association approaches. But recently, I kind of add a new research program, which is one mentioned at, Roman mentioned, is using stem cell and functional genomic approach to try to connect the GWAS finding to the cellular phenotypes that are relevant to the schizophrenia and other neuropsychiatric disorders. This slide, you have seen this slide. The only difference I compare this slide to the slide Dr. Marvin showed is I have 48% here. I don't know where it came from, but so for identical twins, you can see they share the 100% genetic makeup, but only the chance to get the disease is only 48%, near 50%. That means genetic plays a significant role in conferring the sustainability, but also there are other significant players such as epigenomics and also environmental stimuli, environmental risk factors. So at early stage, as I mentioned earlier, when I joined the team at the University of Chicago, at that time, it's a very strong genetic research program. And we try to identify genes, associated with schizophrenia and other NPD. The hypothesis at that time was like, the disease has to be caused by a bunch of genes that at that time called oligogenic model. It's like there are a handful of genes, they contribute large effector site to the disease. So we use kind of a powerful approach for identify those kind of disease, oligogenic model disease, use a linkage approach. Basically, you look at the, affect the same pairs in the family or, parents and proven how they share the, genomic region that confer the susceptibility, that's called genetic linkage. The resolution usually is very low, and the power is not as high as association study, which I will show you later. So at that time, we had, I think this is one of the largest linkage study for schizophrenia. So basically, we use more than 400 families here. Kind of we identify those regions that not call segregate, they are linked or shared by the affect the proband in those schizophrenia families. As you can see, this is like a genome-wide plot. You can see different regions. The resolution is very low, as large regions. You don't know which gene even, it's like a megabase pair region. So this is a region on chromosome 8p, including those genes, as you can see here, like PPP3CC and neuroregulin 1 at that time is very popular risk gene for schizophrenia. So this is the genetic linkage. And then, another approach called genetic association study. So basically here, instead of look at the, shared by affected siblings, affect the same pairs, you look at the, as a population level, you look at use SNP as a marker to compare like between cases and the controls, whether the SNP as a marker, is more frequent in cases or in controls. So you can see here, for example, this is example, you can see if a allele is, case is higher than controls, this is extreme case, in schizophrenia, we never see this kind of case. It's a very minor difference. But this is extreme case, just to show how the association work. So with this, you can do association test to see whether the SNP marker is associated disease in the population if you compare, cases versus control population. Usually at the early stage, the population, the sample is very small, kind of in 1,000. So, the association signal actually kind of stochastic. Sometimes, we identify gene that's associated with schizophrenia, but other study use a larger sample or similar sample, they could not replicate that, which is the case for many of the study, including ourselves at that time. As you can see, at least some gene we studied in the lab, including, one called trisomy receptor at 6q, and neuroregulin 1, that's Manning, and the CUMT, Dr. Alan Sanders and I, we have been contribute to the literature at that time, but later on, it kind of turned out, many of the genes, including the ones, we have tested here, cannot be really replicated if you use a much larger sample. So that means, a lot of association identified, use small sample for schizophrenia research at the early stage, really, is evidence hanging in a balance. It's not like there's no certain evidence. Until, the GWAS stage, you heard about a lot of GWAS, but GWAS, genome-wide association study basically really opened the research avenue for the schizophrenia and other neuropsychiatric disorders, like 2009. This is a trio Nature paper, including one from our group, led by Dr. Gedgeman. You can see, we identify one locus 16p is significantly associated with schizophrenia at that time. Its MHC region contains HRA genes, so it's like immune-related gene clusters in this region. Other locus have also been identified, but a very small number, only 1 or 2 genome-wide significant loci have been identified through this, effort, like combined effort, this trio of Nature paper. So this is the breakthrough at that time. So what's GWAS? We have talked about, genome-wide association a lot today. So basically, similar to association approach I mentioned earlier. So you have, instead of testing one SNP marker at a time, you test them, in the large scale. So basically, you have 1,000 or millions of SNP marker in a chip, and you use hybridization to interrogate those SNP marker, the allele frequency in cases in controls in large number, usually more than 10,000 or even million control samples, and then you do the association test, genome-wide, and you determine a genome-wide significance threshold. Anything above the threshold in the Manhattan plot here would be, genome-wide significant association. So this is genome-wide association. This is really unbiased approach to interrogate the whole genome for SNP markers or loci that are associated with a disease. So with this approach, this is the threshold basically for determining whether a signal is genome-wide significant or not. Why use this? Because in the genome you have 5 or 6 million, common SNPs usually. So you use this kind of threshold to account for multiple testing burden. Now, so with this genome-wide approach, since 2009, we have a trio of Nature paper, and start early 2011, and to last 10 years from 2011 to 2012, actually you can see the genome-wide significant loci social schizophrenia increased from 5 to actually here 287. In 2014, a landmark paper we reported 108 loci. So this is a Manhattan plot just to show you, which loci associated with schizophrenia. This is from the landmark study Nature 2014. We as a group called Molecular Genetic Study of Schizophrenia, MGS, contribute substantially to this PGC consortium. As you can see, there are many high-rise buildings here. The highest one, the tallest one you can see is MHC region actually. This is kind of, there are, as I mentioned earlier, has HRA gene. So it's kind of consistent with long-standing hypothesis of immune hypothesis. When Dr. Marvin mentioned in North Sphere, you have influenza infection, you may have high risk. So that means, in interplay between this genetic locus and the environmental risk factor may play a significant role in conferring disease susceptibility. There are also other, known gene, known, pathway, for example, glutamatergic gene, and also most remarkably, DRD2 actually is one of the genome-wide significant loci. And there are some novel loci that may represent a new biology. For example, MIR137, which I will talk a little bit more in my next slide. So this is the GWAS, right, in 2014. But if you have more samples, for example, here, instead of a PGC3, WEAV3, we have 76,000 cases versus 243,000 controls. You have 287 loci. So with more samples, you can see you have significant more loci that associated with, can be identified to be associated with this with disease, with schizophrenia. If you summarize the major neuropsychiatric disorders, the GWAS result you can see here, up to 2014. Yet Roman mentioned there are not much happening in last year, but actually you can see much more actually genome-wide significant loci identified for bipolar and for major depression. Imagine each loci has many genes. You can imagine can calculate how many genes can be maybe involved in this kind of complex disorders. For schizophrenia right now we still have 287. I bet if you have more cases, definitely you have more loci to be identified. Compared to schizophrenia, bipolar, major depression, PTSD, for autism actually, you only have very few GWAS risk loci. As mentioned by Dr. Cook earlier, probably, ASD, the genetic risk spectrum for ASD may be different from schizophrenia and other neuropsychiatric disorders, more toward neurodevelopmental, dysfunction aspect. And also there are more de novo and rare high risk, high penetrant risk variant for autism, although they share, a genetic makeup, as you will see from this slide. So this is the genetic correlation between different neuropsychiatric disorders versus bipolar in the recent publication Nature. As you can see here, the schizophrenia, major depression, and PTSD, they are among the the disease that share the most with bipolar GWAS association. ASD is the kind of among the lowest sharing spectrum. So this is may explain kind of clinically you may see they have similar symptoms. They kind of share some common pathway or common gene pathways. So yeah, that genetic pleiotropy can explain those comorbidity, clinical comorbidity, I think. So this is the GWAS, what GWAS told us. Now, in addition to the common variant, identified the GWAS as a major part of the genetic risk spectrum. There's another risk, a very important risk factor called CNV. Dr. Cook already mentioned, introduced the concept of CNV, which is basically the large chunk of, genomic deletion or duplication in our genome. So what I show you here is one of the most common CNV that associated with schizophrenia called 22q11.1 deletion, also called DiGeorge syndrome. Among those 22q deletion carriers, about a third of them, they may develop schizophrenia. So you can see although they are highly penetrant, but still it's not like, you have the deletion, you will have the schizophrenia. So the value for diagnosis, is tremendous, but still you have to consider, other alternatives here. The reason for those uncertainty for the diagnosis, prediction of the disease, likely because this, each CNV region expand a large, genomic kind of interval. As you can see here, you have like a 20 megabase pair region. They span more than 40 genes. So any one of those or combination of some of the gene may contribute the phenotype, different phenotype. So you have a spectrum there, a spectrum of phenotype there. So a third of them, they develop schizophrenia. Now, also exome, whole exome study identified those rare risk variant in the protein coding region. They change protein function. Likely, most strong. The strongest risk factor in those exome region is those called ultra-rare coding protein truncating variant, which means they cause protein, like truncated protein, or the dysfunction, the protein will not function anymore, or they may cause the kind of nonsense-mediated decay. That means the gene just will degrade at the messenger RNA level, so that you don't have the messenger RNA or protein product. So that can cause a very strong effect. They are highly penetrant. These are a handful of genes here, but they are extremely rare. The risk variant those genes are extremely rare. So if you summarize the different genetic risk factors, this diagram can describe that very well. For example, here you can see the GWAS loci, the very common. The population frequency is usually more than 5%, or at least 1%. But the odds ratio is very low. It's like a little bit more than 1.0. Most of them, they're less than 0.2. So this is a schizophrenia 287 loci. Now, if you look at the rare variant side, you have extremely rare CNV here. The most common one is 22q deletion, also 2016p duplication, also confers schizophrenia susceptibility and other CNV regions, as you can see here. But they are very rare, and also some, rare protein coding variant that it can change protein function, or cause protein, the lack of protein product. So those risk variants also confer strong effect to schizophrenia risk. So you have two sides of the risk spectrum here. Now, the genetic discoveries during the last decade or more, a little bit more, really provide unprecedented opportunity for us to understand novel disease biology, to really look into how we can, translate the genetic discovery into novel treatment. So the first thing we need to do is to better understand the disease biology underlying those genetic associations. So that's what I have been doing during the last 10 years, called using functional genomics approach. Now, what's the functional genomics? So, as you know, this is the cell. Inside a cell is the basic, building blocks for our body, right? So inside the cell, you have a, genetic material, genetic makeup, which are double-strand DNA wrapped around those histone proteins. And then you have those genetic material can be transcribed into messenger RNA and then can be translated into protein. And those proteins kind of, confer the cellular phenotype that directly relate to, the clinical symptoms. If we talk about the genetic risk variant, basically at the genomic level, has to be translated into protein, which is protein level, and then connect to the phenotype. And in the between, you have transcriptome, basically the messenger RNA that is like that will be translated into protein. So and also you have protein degradation process. You have product degrade from protein and other metabolite, so they called metabolism. So combining this layer of information, a multi-omic, genome, transcriptome, proteome, metabolism, and also they interact with environmental risk factors here. They confer, susceptibility to the disease through a cellular phenotype and then, clinical symptoms. So the whole process basically, studying how genes and their product function to affect biological process and phenotypes can be considered conceptually as a functional genomics. So in order to understanding those disease biology, we need a model system, right? So we knew from years of research, animal model, let's say you can use animal model to model psychotic symptoms. You can use postmortem brain to understand how synapse pruning, reduce the dendrite or reduce the synaptic protein puncta in density in patient's brain. And of course, you can use a live brain for functional imaging, MR, functional MR to study brain connection. But these are models, each model has their pros and cons, as I listed here. But today, my research focus has been, using stem cell derived model. So we use stem cell to differentiate into neurons and also brain organoid, you can call mini brain, and then to study, how the genetic risk factor affect the cellular phenotype that may be relevant to schizophrenia and other MPD. So I'm not sure if you are familiar with the stem cell, our iPS-induced pluripotent stem cell. So this is actually a Nobel Prize-winning this kind of technique. So how you can derive stem cell from a somatic cell, for example, a drop of blood. So basically, you can take a drop of blood or fibroblast of, skin biopsy. You can, using biochemical or some, express some transfer factors, genes, in the cell, and then convert them back to pluripotent stage. We know we human body, let's say we all originate from a single fertilized egg, right? So that is pluripotent. That you have embryonic stem cell there. So basically, the iPSC has the same property as ESC, embryonic stem cell. So from there, you can imagine from stem cell, you can redifferentiate them into neurons, some other cell types. Of course, neurons would be the most relevant cell to schizophrenia and other neuropsychiatric disorders. And then, if we use that disease model, for example, you have a CNV carrier, you generate iPSC from that CNV carrier patient, and then you can differentiate neural model to study how a gene, which gene affected the cellular model that are relevant to the disease, and use that. You can, use that as a model to screening for screening drugs. So which cell type are relevant for schizophrenia? So this is from the GWAS data actually. If you look at a single cell RNA sequencing data here, so each dot here is a cell from the brain, postmortem brain. So you look at their expression profile, you cluster them into a UMAP, T-cell map here, and then you look at the cell type specific expression of each gene, and you look at enrichment, like we talk about GWAS association. You look at those GWAS association in the enrichment of those GWAS signal in each cell types. What you can find actually here is from the Nature 2022 PGC3 consortium paper. You can see here, neurons are most relevant cell type for schizophrenia because schizophrenia GWAS association signal are most enriched in those cell types. But this is not necessarily mean, other cell type are not important. For example, Dr. Marvin mentioned in synapse pruning. The cell type important for synapse pruning actually is microglia. It's the only immune cell, residual immune cell in the brain, right? So microglia is responsible for pruning the synapse, of the neurons. So yeah, other cell type may also play important role, but the most genetically relevant cell type are neurons. So now we have a model. We have a stem cell. We can differentiate the stem cell into different type of neurons, glutamatergic neuron, dopaminergic neuron, or GABAergic neuron. And we can also differentiate those cells into a stem cell into microglia, as I mentioned, important for synapse pruning, and also astrocyte, which provide nutrient for neuron maturation in the brain. And usually, if you in the dish, the culture in the dish, this 2D culture, they are very pure, so that can easily know to study gene function use this kind of 2D model because they are very relatively pure. Of course, there's the limitation because in the brain you have a different type of cells they may interact. So that I will talk about the brain organelle that have multiple cell type that may be better recapitulate the brain cell interaction. So with 2D model, we also can 2D and 3D iPSC model. We can also combine with the another technique, maybe called I'm not sure how many of you are familiar with this, like CRISPR gene editing. Basically, you can add the gene, add the SNP here, use CRISPR technique. So with the CRISPR technique, you can introduce a patient-specific mutation in the healthy control stem cell line, and then introduce to neurons or organoid to study the function of the gene. Or you can also use patient-specific, iPSC, and then correct risk allele to non-risk allele, so that you can compare, what changes are because of the risk factor, genetic risk factor. And in the lab, basically, we do kind of single-cell multi-omic profiling. We can do high-content imaging of those cells, differentiate from the stem cell, patient-specific, or genetically modified, and then we can use the model to identify druggable gene pathway. If a valid model has been established, we can use them for high-throughput drug screening. That's the purpose, final goal of this iPSC modeling. So, with this iPSC-derived neuronal model, the first question we try to answer is, how to make sense of the GWAS associations, right? So we have so many, hundreds of loci. Which gene are really causal? How they cause the disease? To address that question, actually, there are quite a few number of challenges. At least some of the challenge here, as you can see, this is one of the, those hundreds of loci, right? You can see each dot represents a SNP marker. So, and each here is the gene track. You can see, the association signal is, similarly stronger for hundreds of markers, common variant, in the single locus. Now you have multiple genes here. And you remember, for example, if this is the causal SNP, it might regulate a gene, let's say 500 kb away. So we really don't know which gene and which are causal variant, which are risk gene, causal gene here. So that's the challenge. Another challenge is, most risk variant here, they are non-coding part of the genome. So we talked about those highly penetrant protein coding risk variant. They alter the protein function. But most of the GWAS risk variant, they don't alter protein function. They regulate gene expression. That means they change the gene expression. Maybe, very mild change, like 20 to 30% gene expression change. So can we really, detect, the biological function with that kind of, for common variant of small population effect size? So those questions are challenging. Comparing to study, those highly penetrant protein coding rare risk variant. So as first of the, this is one of the first study in the field. We try to use open chromatin as a concept to prioritize those GWAS association variant, disease-associated GWAS risk variant. Try to connect genotype to phenotype. Use kind of open chromatin as a concept. So we mentioned that earlier, like our genetic makeup is double-strand DNA, wrapped around those histones. So you can see in some part of the genome they are more open. So in ATAC-seq, kind of high throughput sequencing technology, you will see the sequencing rate here is show up as a peak that are called open chromatin region. So the hypothesis is that the genetic risk variant under those peak, they are more, they are in open region, they are more functional, they're likely to be functional. So use that kind of approach, we try to prioritize the GWAS risk variant among those 100 loci. So you can see here we can substantially narrow down the likely, functional disease causal variant using this open chromatin approach. As a proof of concept, we also show one of the loci, MIR137 here, we use CRISPR, edit the risk allele, and we observe they can change the gene expression of that gene by itself. MIR137 is a small RNA, and small RNA can also regulate many other target genes, including some glutamatergic receptor genes actually. So we showed it can regulate gene expression, MIR137 itself, and also some other target, and also can change the neuron morphology in terms of the dendrite complexity and also the synaptic protein maturity. So this is earlier study we use this open chromatin approach, try to connect the non-coding schizophrenia risk variant to cellular phenotype that may be relevant to disease. And later on, we kind of use a more systematic approach, try to identify, genome-wide, which of those GWAS risk variants they can affect the chromatin accessibility to regulatory factors. So the approach here, we use ATAC-seq, we call allele-specific open chromatin. So basically, ATAC-seq, if you compare a heterozygous SNP, individual with a heterozygous for a GWAS risk variant, you have two alleles. Now, if you have sequencing rate of one allele is more than the other, more likely to be functional because one allele may be more accessible than the other allele may be functional. So with that concept, we use 20 iPSC lines to generate iPSC, differentiate them into dopaminergic neuron, GABAergic neuron, and glutamatergic neuron, and then we profile the ATAC-seq open chromatin regions, and we called ASOC, allele-specific open chromatin variant, and we studied the enrichment for GWAS association, and also we use kind of CRISPR and single-cell RNA-seq to edit those specific SNP to see whether the risk variant itself can really change the gene expression and neuronal phenotype. What we found was, there are a lot of ASOC, meaning, risk variant, they show differential chromatin accessibility, and they are cell type specific, as you can see here. And this is one of the GWAS loci called VPS45 locus. It has the risk variant you can see in glutamatergic neurons show strongest ASOC, meaning one allele is more accessible than the other allele in glutamatergic neurons. So we used CRISPR editing to edit the SNP. We have so that we can have the iPSC line now heterozygous for risk allele, non-risk allele, and also the heterozygous, one risk allele, another is non-risk allele. And then we differentiate into neuron. We look at the gene expression, which gene affected by the SNP editing. What we found was, VPS45 itself, you can see this here is the VPS45, this is the SNP site. VPS45 was affected. Interestingly, actually, another gene kind of transcribed in opposite direction. It's called long non-coding RNA. That means it does not encode the protein sequence. It's coding kind of a long non-coding RNA. It can regulate other gene expression. So also, its expression was also changed. And interestingly, there's another distal gene, but kind of 100 kb away, I think, also affected by the SNP editing expression. So we think, it's interesting because one single locus, you can have multiple genes that may be involved in regulating the gene expression that may be relevant to the disease. So we test the hypothesis whether those risk genes, the 3 risk genes here in the single locus, have synergistic effect at the transcriptomic level. What we did was we kind of knocked down, each gene individually, and then we did another combinatory triple knockdown. So we compare, if you based on the additive model of genetic risk, you can calculate, 1 plus 2 plus 3 equals, 6. Now, if you have combinatorial triple knockdown, you would expect you have more upregulate or more downregulate. It's not like equal to the additive prediction here. So we can see here there are 1,000 genes more upregulate or downregulate here. If you look at the gene pathway, involved in those more up or more downregulated, you can see they are enriched for schizophrenia relevant ontology terms, like schizophrenia GWAS, MIR137 pathway, bipolar differential gene expression, and the same for the more downregulated gene here. You can see including antipsychotic drug treatment. So that means, this is a kind of interesting. One single GWAS locus, you have multiple genes involved. They can show synergistic effect that contribute to disease-relevant, phenotypes. We also studied the neuronal morphology chains, which I didn't show you here. You can read the paper if you are interested in. So what I'm talking about until now is about common GWAS risk variants. So how we can use iPSC to model, those common GWAS risk variants. It's challenging because, they have small population effect. Actually, in the field, more low-hanging fruit, or more people would like to do is to model highly penetrant, protein coding risk variant or CNV, because they are highly penetrant. From cellular phenotype, if there's any phenotype change, it's easier to interpret. So this is the summary. Basically, your iPSC model, you can model some, mostly related to neurodevelopmental disorder like autism spectrum. But for schizophrenia, bipolar, you can also use them to model, some specific, genes, genetic defect. So in our lab, we also did some exploration on this part. Basically, we have been focused on a 15q 13.3 deletion that associated with schizophrenia. So what we did was, we tried to, because as I mentioned earlier, each CNV region has many genes. They span multiple genes. You don't know which is the driver for the phenotype change, right? So in order to identify that, we did a kind of deep sequencing So here, basically, you can see we did this sequencing in those CMV regions systematically. At this region, we found some nonsense mutation, which cause stop codon that can cause protein truncation or cause nonsense decay of the gene. And then those mutations we found only present in schizophrenia cases. Very small number, but it only present in schizophrenia cases. We hypothesized that this gene may be the driver gene. Those nonsense mutation may cause protein loss of function of that gene, may contribute the cellular phenotype that may be relevant to disease. So we kind of use the similar approach I talked earlier, use CRISPR editing to introduce this patient-specific mutation into a healthy control iPSC line. And we generate the iPSC and then differentiate to glutamatergic, which exciter neurons, and we study their neuronal phenotypes, and also study the gene expression transcriptomic changes. This is one of the phenotype we assay. You can see here we look at the neuron firing in terms of the neuron, how they are more active or less active. As you can see, for that deletion, the mutant allele caused the neuron firing, substantially reduced neuron firing in glutamatergic neurons. So that means this is likely to be kind of the risk allele likely to be contribute to reduce the neuronal activity, maybe consistent with schizophrenia. So this is the one example how we use iPSC to study a rare variant. We also, like I earlier mentioned, we also use 3D. Why we use 3D model, which is also called the mini brain or brain organoid, because they have multiple cell types. The good thing is use 2D. They are homogeneous. You can get very pure cell culture, neuron culture. But in reality, the model that can be better recapitulate the brain complexity is, the organoid, also called the so-called mini brain. So how to do that? Basically, you have iPSC, you grew them in a dish, you keep culture, you use shaker or not shaking. You can let them kind of grow in a group, and at some point they form into like a ball-like structure. If you cut them, you stain them, they look like, they have some kind of, cortical-like structure. You have ventricular zone, you have deep layer, and, upper layer neurons. This is ventricular zone. So it's kind of resemble the brain kind of a cortex structure. So with that, model, we kind of study a highly penetrant protein coding called truncating risk variant I mentioned earlier in the gene called SETD1A. SETD1A is the gene actually modify the histone methylation. So these are the risk variants identified in this gene associated with schizophrenia on the upper panel here. And lower you can see those risk variants that can also cause other neurodevelopmental phenotypes. The most interesting mutation, I think, is exon 16 because it's recurrent, as you can see. Some of them are de novo, but this one is like recurrent here, you can see. And also, enriched in exon 16. Now, so we use CRISPR editing, try to introduce this risk allele into those healthy control iPSC lines. Here you can see we differentiate to we have the CRISPR engineered iPSC line that carry the risk allele and non-risk allele, and then we differentiate to cortical spheroid or organoid. And then we did transcriptomic analysis and also morphometric analysis, and also we measure the calcium imaging with measure neuron activity. This is the confirmation of the how the risk allele can cause reduction of the protein expression of SETD1A. You can see in mutant allele has 50%. This is heterozygous. Or what we generate here is heterozygous. So you have reduction of the protein level. And then you look at the function at the cellular level. You have reduced as SOX2 positive cells, and you have premature kind of neuron appear in the early stage of organoid. So this kind of precursors neurodevelopmental effect associated with this risk allele. Now, a later stage, you can see more like cortical structure, like morphology here. And then, if you look at calcium imaging, which is measured neuron activity, this is individual neuron, calcium imaging, and then you compare that, case wild type, the mutant line, you see mutant line, mutant allele associated with reduced firing frequency in exciter neuron. Now, this is a video shows you how, basically it shows you how the neuron firing. You can see, they are flashing, each cell in the organoid. So how we can use this model? So we didn't, reach this step yet, but, this is example show you how you can use this model to do drug screening or to test, to validate the drug effect. So here you can see they used group from Stanford as a friend. He used kind of organoid cell, the cell dissociated from organoid to measure calcium imaging. You can see this is 20q deletion syndrome carrier. They show reduced calcium spike, calcium signaling capacity. Now, if you use antipsychotic drug, you can see they can rescue the phenotype to the control level. And if you stain those cells, you see they have quite abundant expression of dopamine D2 receptor, which makes sense because they are the target of antipsychotic drug tested here. So, with this kind of model, you can imagine, we can use that to screen, novel drugs, novel chemicals that may be, more effective than classical antipsychotic drugs. So up to now, I have been talking about, how we can use iPS-derived model to use CRISPR editing to edit one gene at a time to study, how a gene affects the function, cellular function. Now, actually, in NIH, we have a consortium called SciGene, try to kind of scale up the assay, try to assay the neurodevelopmental and neuropsychiatric disorder risk gene in large scale. We, as part of the consortium, Dr. Jiping Pong and Rutgers and I, we call that the assay center called MIND. We named it MIND. So we propose to kind of create loss of function allele iPSC lines for about 200 genes in 6 different donor genetic background, and then we differentiate into neuron to study their function, like electrophysiology and transcriptomic properties. So up to, this is as of end of last year, we have 45 genes on 6 donor lines generated, but actually now we have close to 100 genes generated. So this is the paper published Stem Cell Report last year. We kind of summarized what we've done at that time. So basically the pipeline was, we use CRISPR editing to introduce premature stop codon, which is kind of model the, the ultra protein truncating mutation in those neurodevelopmental risk genes. And then we kind of bank those iPSC line, engineered iPSC line, and we differentiate into neurons, different type of neurons, exciter inhibitor, and co-culture them with S glia cells. And we do high content imaging to look at whether, they can change the neuron dendrite or synaptic function. And we also do single cell RNA-seq, try to identify what's the convergent gene pathway, systematically across those, hundreds of neurodevelopmental risk genes. Just to show you some of the results from the high content imaging here, as you can see, SHANK3, you heard earlier from Dr. Cook, is important for autism and cause, reduced synaptic function. And here, indeed, we kind of validate that, use this kind of high scaled approach. And we also, and this is one of the novel schizophrenia-associated genes. As you can see, we found reduced, synaptic dendrite, synaptic function, and also synaptic punctal density, and also the reduced neurite outgrowth, which is, kind of consistent with the reduced neuron function in schizophrenia. So to summarize, basically, during the last 10 to 15 years, through GWAS, whole exome sequencing or whole genome sequencing, we have identified many, thousand common genetic risk variant but they have small effect size associated with schizophrenia and other neuropsychiatric disorders. Another side of the spectrum, we have a rare risk variant in protein coding region that it can alter protein function or, cause loss of function, basically. But they have large effect size. So NPD, represented by schizophrenia, I would say, is most heritable among those NPD. They are polygenic, meaning they have many risk genes, hundreds of thousands risk genes, each contribute a small effect. Individually, they may not, matter, but combining them, in pathway level, they may, they may contribute to substantial to, disease stability. Also, those genetic makeup are shared across major psychiatric disorders, as I showed you earlier. And, the common risk variant, as I showed you earlier, is mostly non-coding. Various, small percentage, they may change protein function. They likely to affect the gene expression and chromatin accessibility. That means they are regulatory variant. A functional genomics approach, using iPSC in combination of the CRISPR editing, as I showcase here, they may help to connect the phenotype versus to genotype, connect the genotype to phenotype. It will help us understanding the disease biology underlying those GWAS associations and also the rare association with the rare risk variant. Lastly, a scalable assay for those neurodevelopmental and neuropsychiatric risk gene underway, which I think they can help identify some convergent, or divergent gene pathways that may be, shared by different psychiatric disorders, and maybe also specific to some, one specific disorder. Those information, obviously, they can be exploited to develop more tailored clinical treatment. With that, I thank you for your attention. These are the people, really working in the lab. Thank you very much.
Wow, thank you. Questions? Rob?
Yep. Could you predict future state of CRISPR gene therapy for psychiatric disorders?
So I'm just going to repeat the question and then ask Jabal to come up to the microphone. So the question was about being able to predict CRISPR and how it might impact psychiatric disorders.
I think, that's a fantastic question, I think. Even for, Alzheimer's, I also study Alzheimer's disease, use this iPS model CRISPR editing. Even for ApoE4 now, people have been talking about to use, use CRISPR editing to correct gene ApoE4 to ApoE3, ApoE, That's, ApoE4 has a very strong, penetrance, basically, kind of more familiar than schizophrenia. So I think it would be challenging for schizophrenia and neuropsychiatric disorders. But for those rare variant, I think, extremely rare variant, definitely is doable. And it's, I think, in terms of personalized medicine, I think it's worth to do. I think it's very promising in the future, I think. But it would be very challenging for common, a risk variant. Yeah.
Super. So I'll ask one last quick question, sort of follow up to that. If you could comment on the overlap between neurodevelopmental disorders, the neuropsychiatric disorders, and neurocognitive in terms of sort of thinking of Alzheimer's and things like that, where you see some of these genetic linkages, and are there any common threads between those three clusters of disorders?
Actually, I think, I'm not expert studying, cross disorder, there's a consortium under PDC, they specifically analyze this cross disorder genetic overlap. But if from the analysis, you do see substantial overlap, across those different orders, different disorders and the spectrum, symptom spectrum. Yes, I think the simple answer is yes, I think. Yeah, super.
All right. Thank you. One of the things that's particularly nice about the symposium, both last year and this year, has been to be able to see the resources within our own department and within Endeavor. So really thankful for that. So with that, we'll go to our keynote presentation today. And actually, I think I have to do something with the slides here. So with that, we'll go to our keynote presentation. David Cohn, who constantly reminds me he's not a clinician, but certainly an entrepreneur and someone with a different perspective. And as we were chatting about what to have him focus on for this presentation, it follows up a little bit on the discussion we had at the end of the last symposium a year ago when we had Dr. Tom Insel here, and really thinking about where technology and particularly AI, which obviously has become a buzzword over the past 12 or 18 months, but where some of these emerging technologies can really help us in terms of health in general and mental health in particular. And so he recommended David, and as we've connected, really excited to see his leadership in this space. And he's going to talk to us about innovation in mental health, lessons at the intersection of entrepreneurship, systems, and values. So David, come on up, please.
I can use that if I use this. All right. Thanks, everyone. I'm going to free myself from behind the lectern. I hope that's okay. I might also ask some questions of you all while I'm talking, and feel free to answer if you want. We'll make it a little bit more interesting, I think. Ramon, thank you for acknowledging that I'm not a clinician. Now I don't have to say it again, although, as you can see, I just did. I am going to mention, though, a few companies across the next few minutes, and I just want everybody to know that mentioning these companies is not an endorsement in any way, shape, or form. As you'll see, some of these companies don't exist anymore, and we're going to talk about some of the reasons for that. So across the next few minutes, what we're going to do is I'm going to start by sharing a little bit of my own story from my childhood, being a patient myself, to a founder. And I would say I'm probably best known for having built one of the early telepsychiatry companies that ended up becoming a national leader. That company was originally called Regroup. That company is now called Array Behavioral Care, if any of you are familiar. But what I want to show in context is how this got built within certain systems and how the values inherent in those systems affected what I thought I was going to be able to do and ultimately what I was able to do or not, as you'll see. And then we're going to talk about a lot of the innovations that are emerging right now and how the systems that they are emerging from and the values within those systems might affect your ability to interact with them or how they show up in your clinical practice. And we'll get into how they sometimes conflict and how sometimes they work together and maybe how you can use them to your advantage as well. And my hope by the end, can everybody hear okay on this mic, by the way? Okay. My hope by the end is that you feel like you have a little bit more agency and you feel like you know how to face all of this change that's happening so quickly right now, because there is a lot of it. This is full circle moment for me because I was born at Evanston Hospital almost exactly 45 years ago. Predated the Endeavor brand, of course, but as we know, all part of the same system now, so I feel like I'm home a little bit. And I was born, I would say, both to an interesting family at an interesting time. You know, I had a very close family member that had a fairly prominent role at the North Shore Health System for a long time, and I don't know that anybody involved in the organization of this knew that when they asked me to do this. but this feels very close to home for me. Also, 2 of my 3 daughters were born at Evanston Hospital. When it came to having resources growing up, I had them. I can't complain about that. But we had a lot of challenges in my family system. And it took me, I should say, it is still taking me time to understand them, but it has taken me decades to understand the implications on my own life. And as I become a parent, and we're going to talk a little bit about that, and to give just a little bit of context, I think that I came from a family that had been in fight or flight mode for 5 generations. I'll spare you all the details, but my understanding is initially there was a real threat, and that threat was escaped, and people got to a safer country. But I don't think anybody in my family actually internalized that they were okay or safe. And that was exacerbated by things like my paternal grandfather fought in the Battle of the Bulge. He helped liberate concentration camps. He was one of the few surviving members of the group that he was fighting with. And these are not things that go away on their own, as we know. Also, around the time I was born, we were moving from a world in which Carter was president, and I would say this was a little bit of a kinder, gentler world, to one in which Reagan was coming into power. And if I had to just isolate one variable that I think is important as we think about this, it was how capital was injected into so many different parts of our lives. And parts that I think before that were kept from it. And that's going to come into play too as we talk about some of the systems today. One of the first memories that I have in my life, I couldn't have been more than 3 years old or so, was playing with one of these nesting dolls. I don't know if any of you had them at home and I very clearly remember being on the floor and taking this thing apart, and I actually found the doll just a few days ago unexpectedly. I have it right down here. And I remember thinking, at least I think I remember thinking, oh, I come from something bigger. I fit into something that is larger than me, and that has impacted me. And oh, there's another doll that goes over that, and so on and so on. And I remember being pretty impacted by this, and I thought it would make a nice metaphor for the talk today, because we live within systems, and those systems live within other systems. And in my life, when I have come up against something that I don't understand, it usually means there's a bigger doll with some bigger lessons to learn coming in over the top that I have not understood yet. So we'll come back to this. But the very first bumping up into the systems that I confronted was my family system. And as you can probably imagine, as a kid, you think that's kind of all there is, right? You live within the family system, and that's your whole universe. And because of the aforementioned trauma that was not dealt with, I had a real felt sense that whatever I needed to be okay as an individual were not being met by the family system that I was living in. And I know now that was no fault of theirs. But that manifested in anger and sadness. And these were things that were not acceptable emotions to have in my family because they made us feel unsafe. And I think the only thing scarier as a kid than feeling like you aren't okay is that your parents aren't okay. And so what I did, which I think many of you will understand, I feel like I'm in a safe audience here, I turned it in on myself and I internalized that. And I said to myself, I am a bad person. There is something wrong with me, clearly, because the idea that there's something wrong with my family system is unbearable to me, because that would be far worse. And that came out in pretty spectacular ways. When I was about 12 years old, I lit my street on fire so large that the fire department had to come. Yeah, I can laugh about it too now. I was not laughing about that for quite some time. And it was only a year later that I started using drugs. And it wasn't long after that I was using drugs every day. And I have a really good friend, a social worker named Lauren, and she says, you know, I am sick of treating fish in toxic waters. We don't have a fish problem. I'm sorry. We have a water problem. And it took me a very long time to come to grips with that I had a water problem in my family. And I think that's really common. And I think we got some water problems in society too. But to my family's credit, I did get care, and that changed the course of my life. And I'm very grateful to people like you in this room. It's actually still emotional for me to talk about it. And this was where I think I really learned how to interact with other humans, really, to see it modeled. And it had a huge impact on me. And at the time I wanted to be a clinician. My two older sisters became mental health clinicians. And I really credit what happened in therapy. And of course, you know, I'll always be in therapy. I think it's good mental hygiene, and I feel like I'm always improving and learning. But I attribute so much of my ability to turn my life around to what happened in those sessions. And I'll just share that 3 of my best friends from that period didn't make it to their 25th birthday. There were 10 of us. 3 of us didn't make it past 25. And so, as you can imagine, I still had a lot of work to do. I still have a lot of work to do today. But one of the things I discovered was that I had the ability to achieve if I set my mind to it, and that I could get some external validation and feel good about myself if I went out and I did impressive things. So I went on sort of a tear of trying to do impressive things and impress people, obviously with the ultimate goal of trying to feel good about myself, with varying degrees of success. That took me 2.5 years to the Peace Corps. That took me to work with C-level executives in companies that were the largest in the world, all over the world, where I got to learn from very impressive people about how to build things at scale. And what I learned from those executives was they weren't smarter than any of us. They just knew how to simplify problems and surround themselves with smart people who fill their own gaps. And I thought to myself, well, I could probably do this. I'm going to give it a shot. I went to business school. And as I had mentioned, I was really passionate about mental health, and my other passion was technology, and I'd been tracking technology to have real-time live video interactions in the web since the 90s. So I was really early into that. And I thought, well, it just makes good sense for us to build a company that tries to solve mental health access challenges by using this emerging video technology. It was finally getting good enough at when I was in business school, around 2009. And so that's when I started that company, and I built what became Regroup. And I thought I was building a tech company when I set out to do this. And I thought that the value was going to be providing bells and whistles for clinicians so that they loved using this thing, and that patients would just magically appear. And this is sort of an early version of what it looked like. You can see the 2 video feeds in a browser. And it took years to figure out how to make this thing work. And what we discovered finally was that in Illinois, within Medicaid, if you had a patient who had their feet in what we call a qualified destination site, Medicaid would reimburse for the service. And so I found a few friends who worked in community behavioral health centers and in health systems who had large Medicaid populations, which was exactly where we wanted to focus our efforts. We really, remember, I was a returned Peace Corps volunteer. I was trying to help people if I could. And they said, man, we have not been able to recruit psychiatrists here for years. We are way understaffed. If you could bring some psychiatrists via video and we can get paid for it, even though we're probably still taking a loss on Medicaid, we'll do this all day. And I said, okay, well, how do we, anything else you want? And they said, yeah, put them in the medical record and let's credential them so that they're an extension of the team and they look and feel and act like they're part of the team. And I didn't have any preconceived notions about this at all. And I just said, well, it sounds like we should probably do what you want us to do. And so we did, and the thing exploded. I mean, after 3 or 4 years of trying to get this off the ground, it went from 0 to 100 in 10 seconds flat. And we went from seeing 0 patients to, in a period of a month, thousands to tens of thousands, to then, in a period of a year, hundreds of thousands. And by the time we merged, which I'll get to, we had crossed into the millions of patients. And it was really based on this simple idea of integrating the clinicians into the local care teams and making sure that they could get paid for within the systems that they already lived within. And there had all these people at that point trying to do things in ways that were complicated and fancy and didn't work within the systems that they had come through. And so I, like many people of my generation, thought, well, this is cool. I should go raise some venture capital, and we should make this much, much bigger. It seemed like a very good idea at the time, I have to say. And that's what I did. And I thought that I had won by raising millions of dollars from top-tier venture capitalists. And for a few years, it felt like I had, because they gave us a lot of money and they gave us a lot of leeway, and we were able to really accelerate this into a very large company. But what I didn't understand about venture capital, I'm going to explain to you, which is, I think, our first lesson on understanding the systems that we live in and the implications on us. Venture capital is the riskiest type of capital. People invest there because they are looking for a grand slam, and they're willing to accept big risk. And the way that VCs make that happen is they encourage their portfolio companies to take very big swings at every pitch, if you're a baseball fan. What else happens to people who try to hit a home run every time they swing? Yeah. And VCs are okay with that. That's their whole strategy is, 6 of you guys aren't going to be around in a year, but the other 3, they're going to make us our money back. And all we really care about is the return on the invested dollar here. That's what we're in business for. And by the way, a lot of these are very good people. It's not that they don't care, but investors are there to make money for the people they invest for. Now, when we raised the money back in we raised a few tranches of capital. When we started doing this, 2014, 2015, interest rates were around 0. Money was everywhere. And I wouldn't say it was easy to raise money. They had a lot of cash, and they needed to deploy it. What I didn't know that ultimately created a big change in my life was as they felt interest rates were going to start to go up, their strategies had to change. There is an inverse relationship between interest rates and invested capital and venture capital. And when venture capital gets involved in your company, they take over your board. You're essentially giving them control. And a board of directors in a company like this has one job: hire and fire CEOs. And I had started this company with the idea that we were going to bring access to the people who had the highest level of need. And that was largely in the Medicaid population, and that's what was working for us. And we had built a whole culture and a whole mission around this. And I very clearly remember the board meeting where we sat down and they said, "You're doing great. We just need you to really juice the profitability now." And I said, fee-for-service reimbursement rates in Medicaid are fixed, and there's a market for talent out there. And they said, "Well, we're not telling you to abandon Medicaid. We're just telling you to get more profitable." You can imagine what happened. There was no good way to square this one, and it just didn't align with my values anymore. And it took about 2.5 years before I actually left the company. I know there was a controlled merger, and that company that we created, Array Behavioral Care, it's doing good work. It's doing good work, but it wasn't the right place for me anymore. And that was a very clear lesson to me about how not understanding the systems that you're in can have a pretty big impact on your life. You know, I thought I was doing one thing, and next thing I knew, my entire identity was shredded and I had no job. So my hope is that if we walk through some of the systems that you might encounter together and their values, something like that doesn't happen here. And perhaps it isn't that anyone's losing jobs, but perhaps it's losing agency right now as technology is coming out. And I want to get into that as we move into the second half. So we've already talked about this VC doll, and what it really cares about is profit and scale. Investors invest to maximize returns. That's why they do it. Again, they're not bad people. They should be subjected to other systems that keep them in line and do healthy, productive things. And that's where we have the other dolls. You have a payer doll, and to do anything at scale in our society, we really need to play nice with this payer doll. It could be $3 or $4 if we wanted to get into it. We won't do that today. And they're really trying to make sure that there are enough resources to go around for the risk pool that they're trying to cover, and that whatever's being paid for is based on good clinical evidence. We have an EMR doll. We could have called this system health system infrastructure if we had wanted to. This one is really important and interesting, by the way, and I think sometimes we don't understand the importance of the role the EMR is playing for us. And if you go back and you want a riveting history of this, there is a 4-hour podcast that's just come out on the history of Epic. It's fascinating stuff. But they really were formed to ease billing and to make sure that everything was done securely. And then we have a governance doll whose job it is to keep us safe. This is the FDA, this is the DEA, state regulatory boards. And then we have all of you here today who are the flag bearers of quality and coordination. You are where all of these things are supposed to come together and move through you as a clinical service to your patients. And we are by no means going to be exhaustive when we talk about the following items, because any one of these could be large volumes, and they're moving so quickly that I was getting into bed last night and the Wall Street Journal published something that changed some of the content today. That's how quickly these things are moving. We'll start with telehealth, we'll move through AI. I want to talk a little bit about wearables and the consumerization of health, digital therapeutics, and if we have time, I'm going to talk about my second attempt at entrepreneurship in the mental health space. This company is called Sanarte. So I think moving into telehealth, clearly the most impactful thing that's happened in the last 10 years was COVID, right? COVID changed the world for telehealth. And it was interesting to be in the space before and after it, because before it, people would laugh me out of their offices. I mean, plenty of people were using it, but people would still literally laugh at me. And people don't do that anymore when we talk about telehealth. And the second most impactful moment for me was when Epic created their own integrated module within the EMR so that clinicians could provide telehealth from within the health system in a way that played nicely with billing, with coding, with scheduling, because before that, it was the Wild West. And I want to say that this is a good example of a distinction between when you bring something within some healthy systems, and trust me, I understand that there's plenty to complain about when we talk about Epic, but it also does a lot of things right, and it creates a lot of safety and security in terms of using these technologies. And I want to compare that against a company who, well, there were a few that you all probably know. They raised large amounts of venture capital, and they decided that they were going to stay outside of the payer system, and they were going to stay outside of this infrastructure of the health system, and they were going to create direct-to-consumer telehealth companies around specific conditions. What happened? I mean, does anybody remember those stories? I'll tell you what happened. They did $100 million in very short order by prescribing 40 million pills inappropriately of Adderall. And the CMO and the CEO went to jail. And because they weren't subject to these other systems, it took until the governance doll woke up and said, what the heck is going on here? Because the other dolls weren't acting on it because they were trying to live outside of that system. And the only prioritization that was happening was around profitability. So when you see Epic build a module and bring it in-house, you do have to stop and say thank you, because they are creating some safeguards there. Now, just very quickly, we're going to go through the state of telehealth right now. It obviously peaked during COVID, but almost 30% of every mental health visit right now on all Epic systems across the country is a telehealth visit for mental health. Are you all providing telehealth here? You are, correct? Yeah. Within the last day, last 2 days? What? All the time? Okay, so 30% seem about right? Higher? Okay. Okay, yeah, not surprising to me at all. So this is nationally, and I spoke to the CEO of a large payer last week, and he said across the payer, they were at about 45%. So you guys are high, which I think it speaks to the savviness of you all and the consumers. But mental health as a specialty uses telehealth at a way higher rate than any other specialty, including primary care. And it makes sense, right? And the thing that stands in the way really of greater rolling out of telehealth is not technology at all. Again, once Epic is building something into the EMR, it's commoditized. There's not a technology play anymore. And for the purposes of this conversation, when I say telehealth, I mean synchronous video between one clinician and a patient. What is holding us back a little bit are the state and federal rules. So they keep us safe, but we have more than 50 state licensure boards, if you count the Virgin Islands and Guam and Puerto Rico and all of them. And there is just a patchwork of rules between them and the DEA that is still creating confusion. And especially for the MDs here, if you want to practice in all 50 states, you have to carry almost 50 licenses. I do have to say that the Nurse Licensure Compact, SIPACT, and the Social Work Compact that are coming are trying to do this in a better way, where you have more of a passport than having to get licensure at every state. But the MDs still have to pay for all of those licensures. And the compact that they're in allows them to fast track it a little bit. We use this thing extensively. Sometimes it's great, sometimes it's not. And there are some good historical reasons why we have all these state licensure boards. But one of the not so good reasons is them protecting their revenue occasionally. And I just, I want to call it out because I think it creates inefficiencies. All right, now we're going to move to AI, and I split this into two parts. One part is in the back office, one part is facing patients, and I think there is quite a difference between the two. I'll be curious in conversation with you all to hear what you think. I heard a rumor that some of you are using ambient listening during your sessions to help with charting. Is that true? Can I just see a show of hands who's doing it? Are you guys using DAX, Dragon, Copilot? Abridge, okay. Very similar in terms of functionality. Is that natively integrated with Epic? Yes. Awesome, okay. We frequently at my last company, where we employed a few hundred clinicians, would ask what the largest dissatisfiers were, and no surprise, it was the time spent in the chart. And it was a feeling that that time would be better spent working with patients, or perhaps resting too, because the hours that they were putting in were pretty heavy. So I think something like this could be a real game changer, but it's also coming at us in different flavors from different systems. And I think understanding that can help you decide where you are with your own risk tolerance relative to the sort of functionality you get. You have Abridge and Dragon Copilot from Microsoft that used to be Nuance that has been working on things like this for decades, ranging back from the early transcription services. And you have that protection of it being safely embedded and integrated into Epic. And you better believe if Epic is allowing that, they have some very high walls in their walled garden. It has been vetted extremely thoroughly. And then you have other players in this space, and there are a lot of them. There's a fairly well-known one called Elios Health right now that is really interesting in terms of functionality. But they're venture-backed, so you know that they're under that pressure to perform economically, and that's not always a bad thing. But they also don't have that level of integration. But they do provide some functionality that my understanding is at least Dragon Copilot doesn't. When it comes to especially talk therapy sessions and very unstructured data. And I think they're starting to get into listening to the cadence of the voice. They're starting to listen to syntax. And they're starting to be able to give coaching specific to behavioral health services that a Dragon just might not be there yet. And so I think you're just trying to balance right now whether you're prioritizing privacy and reliability and security, or you want the latest and greatest, knowing that VC lever might push them in some strange directions. And then we get to where I think is a real dividing line for us. And by the way, I think everything you said about artificial intelligence earlier is spot on. When we're talking about this in terms of research, this is a very powerful tool. But there's a big difference between using it in research and unleashing it directly interfacing with a patient. And this is the point at which you start to see the locus of control move away from you all and towards something that's automated, and you have to ask yourself, are those doing it because you all have an expensive hour, and this is a way to be cheaper only, or because it's going to be providing better care? Or where is the balance point? The best known direct AI solution right now is it's called Wobot. Has anybody used it or seen it? There's another one called WISA, and they have taken significant amounts of venture capital. They started in 2017, and VCs usually want their money back in 10 years at the longest. And so they're getting up there, and they have not yet found product market fit. And so you're starting to see some changes. And they have a direct-to-consumer version of this that is not FDA cleared. So it falls in this gray area of support, wellness. The issue, though, is that people who don't have access to anything might go and use these services. And Wired Magazine did an article on them a few years ago, and they were playing with it. And they said, hey, Wobot, I'm going to go walk up to the top of this cliff and jump off. And Wobot said, it's great that you're going to go get some exercise. AI is advancing very quickly. I am sure we're going to get to the point where these things are safer. We still need a human in the mix. And we still need, I think, a clinician in the mix. And I think what you want to look for when you're determining whether you want to use something like this is whether these companies treat you as somebody who's the quarterback in control and they're adjunctive, or whether they're saying, we can do it all. Because right now they're going to employers and they're saying, we're going to replace your EAP programs. You don't need a clinician in the mix. Some of them are saying, well, maybe you should. I just think we got to be a little careful. It's happening, okay? But we don't need to rush into it before we're comfortable with this. All right, now we're going to move to wearables and the consumerization of healthcare. I'm going to give just a quick example, and there's a little bit more in Q&A I'd like to get into in this, but I think this bucket is really large. I would say it could cover everything from 23andMe to direct-to-consumer, clinical tests, to wearables. We're going to focus mostly on wearables. I do just want to say that 23andMe as well, who again took some outside capital, decided that they were going to work largely outside of the system, have now gone bankrupt. What happens to all that data? Yeah, we don't know. We don't know. They have my data. I mean, so it's just something to be aware of. Now, when we talk about wearables, whether it's Fitbit or Oura or the Apple Watch, it is pretty amazing what is happening here. And I'm actually going to just go quickly to this slide, and then I'll come back. In 2024, across the world, there were almost 600 million wearable devices sold. Okay. How many of you have patients who come in and talk about data that's coming off of their watch? Anybody? Yeah? Okay, that's a lot of you. Well, if you notice the lower line here, this is being pushed further and further into society by moving the price down. That's what's happening when you see profitability not going up, but units sold skyrocketing. They're trying to get as mass distribution of these devices as they can. And I actually think Apple is one of the more principled tech companies. But even Apple, and by the way, we could have a really interesting conversation about their architecture relative to their own values. It's actually fascinating stuff. But Apple is collecting, by their count, 150 pieces of data that's coming off your wrist. 150. Some of those are self-input, but most are just coming off your wrist. Take a guess at how many of those are FDA cleared for actual medical treatment. 2 or 3, depending on how you count. 2 or 3. They have FDA clearance for the ECG and the detection of AFib, and they have clearance for early detection of sleep apnea. That's it. And I have to tell you that I sleep with this thing on, and there are days when I wake up and I feel really refreshed, and then I look at my sleep data. Oh, I'm 2% in deep sleep? I guess I'm tired. And we were just drowning in this data here, and it is fascinating that we can have a steady stream of data. Maybe we need more than just our yearly physical checkup to get this information, but I worry too that we don't know what to do with it, and that the average consumer doesn't know the difference between an FDA cleared device and one that isn't, and will misinterpret this wellness and fitness data as something meaningful. And that again is where it would be great to have them integrate into the systems that you all work with and have access to some of this so that you can be the quarterback again. Now I will say that every 6 months, Apple asks me to take a PHQ-9 and a GAD-7. They do. And they ask me to log my mood, which I do. And if I am logging negative mood for too long, they will notify me. But then I have no idea what to do with it. No idea. There's a link to the suicide hotline, but that's about it. And I just think that these totally passive devices without sense making in a structure that is so clearly geared towards profit, I don't know. We got to think about it. And I love this stuff. I use it. So then the question for me naturally was, well, was anybody or is anybody trying to take all of the ability we have in the digital world and move it back into the systems that kind of keep us safe and are vetted? You know, those that are FDA approved, those that maybe payers will reimburse for, that treat real disease. And the answer is yes, we call them digital therapeutics, but it's also kind of a sad story because what you have here, and I'll start with the example of Pear Therapeutics. Pear is kind of the granddaddy of the space. I don't know if any of you have heard of them, but they've raised over $400 million. And they were able to achieve FDA clearance for substance use disorder, opioid use disorder, and chronic insomnia. And as they were doing this, remember how the interest rates started to go up and messed up my business? Same thing happened to them. And this was just at the moment that they started to try to convince the payers that they should reimburse at rates that would help them stay afloat after having taken all of that capital. And the whole thing came crashing down. The payers said, this is cool, I get the FDA clearance, but you need to show me that the difference between other FDA cleared interventions and your digital intervention are good enough to justify all of the money you want me to pay you. And they said, we think it's fine, but there's a lot cheaper alternatives out there, so we're not going to give you these massive reimbursements that you're asking for an intervention that is completely digital. And so after having a valuation of over $1 billion, Pear Therapeutics ceased to exist, and that progress was lost. Hundreds of other companies have tried to walk this path. One of the ones that is seeming to have some success right now is a company called Freespira. I don't know if you've seen it. They use a breathing device and an app to track and help people modulate breathing for panic disorders and anxiety attacks. And the FDA just provided clearance last week for adolescents aged 13 to 17. But they're struggling to get it paid for, again, right? This is where that payer doll is saying, I don't know. And then they have that VC weight on their shoulders to get it done, or else the cord's going to get pulled. And if you don't have the capital, I don't think you can walk that path anyway. And so it's an interesting world, and a lot of these folks are trying to do it in a way that's adjunctive to clinicians and interfaces better with you all, moves data back into the EMR, stays safe and tested. Because I know the topic of the day is schizophrenia, I did look and see what was happening in this space. I could not determine whether Abilify MySite is still on the market. Has anybody prescribed this? Has anybody even heard of this? Yes? Okay. My understanding was that the FDA cleared it by saying it seems not to pose any danger to patients. However, when it comes to medication adherence, which is what it was trying to do with a patch and an app that talked to a pill, we're not sure it's improving adherence. You guys would be in a better position to comment on that. And so it was very hard for me to find data on this. The only other digital therapeutic I found a reference to specific to schizophrenia is called CT155. It's something that Boehringer Ingelheim is working on. It looks like it's bringing in passive data from the smartphone, trying to help people with social interactions, from what I could gather, and give some coaching. It sounds great, but it sounds like it's in the very early stages of approval. See if I have time for this. All right. So, as we go through, or we've gone through everything today, and I go kind of back to my family of origin, one of the things that when I, through Regroup and later Array, was providing mental health services at scale, didn't see was a recognition of the importance of the family system. And I really feel like had my family had support as a system, that we all would have recovered more quickly, and I think we'd have better relationships to this day. And I think this hyper focus on the individual, it was enough to get my life turned around, but I don't think it was optimal, at least not for me. And I don't want to over index on one, but in talking to family therapists and in looking at disease states like eating disorders, where FBT is really part of the clinical evidence base, it seems like there is more we can do with treating family systems and making sure we're supporting every individual and the whole system. And that's what my second entrepreneurial attempt in the mental health space is focused on. And I could not figure out how to do this within the existing payer model, but I also knew the risks of taking venture capital too early and trying to do this. And so I stayed away from that too. We're working very slowly. We're working very intentionally. Our clinical director used to run the eating disorder program at UCSF. And my hope is that we can prove that the outcomes for families are better within the model that we're trying to use, and then we can expand out into broader parts of society and ultimately get this reimbursed. We're not there yet, though. Of course, we're still subject to DEA and state licensure rules. And as we get towards the close here, my hope, I think, is that not even that you think these are exhaustive, all of the systems out there, but just when things start to get confusing, you can step back and say, okay, I'm always getting deeper with the microscope into what I'm working on. What happens if I sit back and I look up and around? Is there a big doll coming over me? And what are the implications of that? What can I do to keep agency and keep myself at the center? And how do I feel about that? What are my values? Are they consistent with where I think things are going? And if not, what can I do about it? And I don't think that we can avoid conflict as we try to tackle these questions. And I think part of what's great about, at least my experience with the mental health system, is it helps us navigate conflict. It helps us navigate conflict externally. It helps us navigate conflict internally. But I hope that you engage when you see these AI tools coming, when you see technology coming, when you see the hottest new venture-backed company that everybody's using, that you stop and think, well, hold on a second. And I hope you engage on your own terms. And, I have to say that, again, in gratitude to clinicians everywhere, I now have a family, 3 kids, who enjoy a much more stable home than the one that I grew up in. And there's something my wife and my daughter say before they go to bed every night that I thought I would share, that it helps me maintain balance, because I live in this stuff, and I find that it can really knock you out of equilibrium. And I often feel like this is a lot. How do I make sense of all of this? How do I maintain balance? And what my daughter and my wife say, my wife is a native Spanish speaker, is me amo, te amo, which means I love myself, I love myself, I love you, I love you. And I thought it really encapsulated what I hope for all of you, just finding this balance of showing up for yourselves so that you can do this work that you do and show up for others, without forgetting that the showing up for yourself part is half of the equation. And I would argue that it's the first part. So this is just something that I keep in my mind. If I'm doing a meditation or something, I often will say that to myself. And I hope also what we've discussed helps you all take some control and make sure that these tools and innovations are working for you. Everything we talked about today is automation, it's digitization. And yet, for me, the most important part of the work that I did was learning how to be with another human. And the only way that that can happen is if you all are present doing this work, okay? That's the only way that that human connection happens. And I think that's the most important thing in the world. And so for that, I want to say thank you for the work that you're doing, and I hope that this can be helpful for you in some way.
And David, maybe you just hang up here for one second, just in case there are any questions. I'm going to take chair's prerogative and ask the first one, which is, given events over the past several months in Washington and thinking of that governance dial and the safety dial that you had on there, what your thoughts are on what the future holds, at least for the next couple of years. If you can try and go into that landmine, I'd appreciate it.
Well, I'm going to start with some easy stuff, which is there has been some progress in recent months making some of the COVID changes permanent around telehealth. So, for example, we were able to get a permanent exception on originating site requirements within Medicare, which is a big deal. We can go into that later. Man, I wish I had a crystal ball and I could say I would be lying if I knew what was going to happen. I don't think anybody knows. And I have some hypotheses, but I don't know. Man, it's tough. It's tough out there. Sorry to dodge. Yeah, I can't.
Thank you for the good talk. I had a question maybe since AI is moving so quickly, I think it's hard for most people to kind of keep up with what AI does and doesn't do over time. Like AI 6 months ago was not the AI now. Do you have a advice or two-liner about like, okay, this is AI, this is what it can do, this is what it can't do?
And David, I'm going to ask you to repeat the question, please, for the folks online.
Okay, so the question is about, with the speed of change in AI, determining sort of what it can and can't do right now, and maybe the implications of that.
Well, like, I've seen it either way. Like some people kind of say, "Oh, it can do all of these things."
Yeah.
It's going to be able. It does answer every question correctly. You got a patient.
Yeah.
Into it, and
Yeah. So
puts all the stuff into it. It hallucinates all over the place.
So here's my one person's take, and I think I know enough about AI to be dangerous. I'm not the world's expert on it by any means. First of all, if people aren't using it in a protected capacity, they're going to go to ChatGPT and do it, right? And this is where it gets so weird and difficult, because like Apple, you're making something available that's sort of pseudo healthcare. It has devoured all of the text. It knows the tenets of CBT and DBT. And, you want a Freudian approach? How about Jung? Sure, it's all that you're right. Like, it's all there. The problem is that when I should say, when we moved from what we used to call machine learning to deep neural nets, if that's meaningful for anybody, my understanding is we went from actually being able to pinpoint what the system and the mechanics were doing to a place where we could no longer actually understand how the outputs were being determined, other than, well, we've ingested insanely large amounts of data, and here's your answer. And I have heard people who've been involved in the development of this, and many will admit the same, that they don't know, and they often can't pull back the curtain to see what computation led from point A to point B. And that, now they can build limited models where they can control more of that, but in the largest models, like ChatGPT, they don't really know what's going on. So I think perhaps the answer right now is to have some clinician oversight, hopefully, and make it adjunctive, and to work on some specific models. So I'd almost rather see you all using more limited models, but where you know exactly what's gone in, rather than it's devoured everything that's ever been written over the last 100 years. I don't know if that's helpful, but because they do still hallucinate, though. Absolutely.
John, make this the last question.
Oh, yeah. Just real quick, I was curious about healthcare delivery systems, and you were mentioning burnout when it comes to documentation. I totally get it. But another big thing is dealing with prior authorizations and payer issues. That seems that it's getting so bad that it seems to be I have to almost like make clinical decisions based on that, and all the labor and cost involved with that. Is there a way to deliver healthcare more directly and cheaply to those that can afford healthcare in a way that avoids the payers?
Well, so the question started around clinician burnout in terms of charting and use of the medical record, but then expanded into needing to get prior authorization, other administrative tasks specific to interfaces with the payer, and if there was any hope of getting out of that. I think the answer is yes, and there's a whole branch of AI called agentic AI that's coming right now. And in fact, I expect that Dragon Copilot through Microsoft, and I'm sure at Bridge as well, will start making suggestions on process steps behind the scenes, and ultimately might be able to do some of the prior auth work. As of right now, I think they can do things like they can sit on the hold time for you and then notify you when a human picks up. It's something. And I think they'll get better. And then there's a whole other set of companies that are trying to work through more streamlined prior authorization specifically. I have some friends who were working at a company called Cohere Health, Cohere. But yeah, I think we'll get there because
I heard the term sludge. I was listening to a podcast about it, and that the idea is that some of these payers are volitionally making it so difficult. You have to like fax something in or snail mail something that obviously doesn't need to be, in the hope that you give up, and that keeps costs down for them.
Are there any payers in the room here? So I think we're on a pendulum. And man, my real views on this are quite unpopular in the world in which I live, so I often don't share them. But oh boy, it is really tough to run healthcare the right way when there is this strong profit motivation. And I know a lot of people in very high positions at many of the large payers. They're all good people. And this system that we live in wasn't really built by them. There are some, one day we should all do a history lesson on how we got here, because it's pretty interesting. And now they are tasked with trying to keep their companies profitable while trying to do right by patients and make sure that the risk pool covers the care that they need to produce in a world where care is constantly more expensive. People are living longer. Drugs are more expensive. It's hard for everybody. But are there tricks sometimes? Probably.
And a huge part of the problem in that system is that with most of the payers, the customer is not the patient. It's the employer, it's whoever else. And so that creates, these sorts of points of contention that really make it difficult to provide good care. So with that, we're going to take 10 minutes to go grab lunch, take a quick break, and then we're going to come back for the panel and more Q&A and discussion. So let's be back in the room, let's say at 12:21 or 12:15, 12:25 or 12:26. Thanks, everyone. There we go. We're going to go ahead and we're going to get started, guys. So if I can ask the speakers to come up and have a seat over here. And Ed, if you can turn on your camera so that we can involve you with the Q&A, that'd be great. And David, if we could have you come up to the stage. Super. All right, so appreciate everyone hanging in. We had a lot of amazing presentations today and really remarkable depth and breadth of various topics that sort of all came together in some way. I'm always amazed at sort of that natural synergy. And first question I have for all of you, what are your thoughts on therapist job security due to the increasing number of people turning to AI technologies for therapy?
Here we go. Just because coming off the presentation, I think if we do go that route, we stand to lose the most important part of therapy, which is understanding how to get along with other humans. And also the most enriching part about being alive. I'm sure many of you are familiar with the Harvard study of adult development, which basically says those of us that are the happiest and feel the healthiest are those with the best relationships. And if we can't get that modeled with you all, I think we're in trouble. So all I'll say is I hope that does not happen, and we need to protect it.
Not going to happen, I don't think. I think AI could be a reminder system, hey, I see you didn't sleep well in the last 3 days. Maybe you should do something today that might be restful. Let's plan that out. But that's where I think it's gone now.
Yeah, it's interesting. So I was involved in the chair searches for radiology and now pathology. And in their departments, for instance, AI is critically important and is going to be playing an ever-increasing role. So I think if anything, right, the radiologists and pathologists probably have to be a little bit nervous in terms of it. I mean, it's framed now as improving access, and there's a workforce shortage, so why don't we try and prioritize things? But I think as you start looking at a chest X-ray, how much of that could be learned by a computer and sort of focus on those things. So it'll be sort of an interesting development in the future. All right, the next question I have, and I think we're going to have someone around with a microphone there, so if there are folks in the audience who want to ask questions as well. But I will, the second question we had online was, how are providers treating patients in the residual stages of schizophrenia? Is the focus on positive symptoms or more on cognitive negative symptoms? And how do you make that a priority? So Rob, maybe you can take first stab at that.
Yeah, so residual phase of schizophrenia is usually in people's 40s, 50s, and on when they've had maybe multiple episodes, generally are doing kind of more stable with maybe a persistent lower level of symptomatology. I think it depends on the patient. In generally the medications are the same choices, so using what's most effective, change if you need to. And then kind of at the end of the talk, I mentioned some kind of non-medication interventions. So if there's certain types of therapy, we didn't really talk about it, but there are some social skills training specifically, or cognitive rehabilitation interventions that can particularly help patients with specific deficits that you notice. The challenge is it's very clinician driven and a very clinical decision rather than it's algorithmic, I guess.
All right, just checking to make sure there were no other questions in the chat. Questions here in the audience as we interrupt your lunch. Jill, hang on one second.
No, just thinking about what you said, Rob, about the incidence of marijuana, how it affects schizophrenia, and we have so many patients that live in their parents' basement and vape all day long, and it just, I don't know, the society just seems to make marijuana so benign, like this idea, and it would be nice to see if there was a more of a push to get that out there.
Yeah, I'm sorry, go ahead, bro. No, I was just going to say, I mean, it's interesting, right? This whole discussion around legalization of marijuana, and particularly as folks have tried to frame it as a medical marijuana, right? I always remind people it's medicinal, not medical, because all of the professional medical organizations are against the legalization of marijuana. There's not one that's in favor of it, right? In part because we can't predict who is going to be vulnerable, right? So I always think back to, as I date myself, the past times at Ridgemont High, right? And think of that subset of people who just are not productive, not able to move out of their parents' basement and do all those things. And looking at a kid, I don't have any idea of who can smoke marijuana fine and, who can't. And so with that, it's such a high risk, sort of low reward sort of scenario. You sort of wonder why it ever made it. And I guess the answer, and I'm sure at least David would probably agree with me, right? It's money that drives that, right? In the same way, why are cigarettes legal and alcohol legal, right? Because without it, the state of Kentucky would disappear, and Milwaukee would, and St. Louis would disappear, right? So these are just economic decisions sometimes that are not driven by clinical decisions. So Rob, I don't know, anything else you'd like to add?
Yeah, it's a big challenge because before it was legal, you could just say it's illegal, so you shouldn't be using it. And that was an easy out for us as clinicians. Now that it's legal, I guess my tact on it is, yes, it's legal, but it's not a good substance for you. Like whatever it just doesn't work with your body. It doesn't go well with your body. Every time you've used, you've ended up in the hospital, and use kind of that motivational interviewing strategy to kind of help raise up the level of consequences to their awareness enough so that they'll be motivated to make a change or cut back or whatever they can do to reduce that risk. Yep.
Super. So Ed, I appreciate you being back on mic, and you were started us off this morning. So I'm wondering if maybe after listening to any of the speakers, there were any questions that you had that we could take advantage of. No pressure if not. Hang on a second, Ed. We lost the sound here in the auditorium.
Sorry.
Oh, there you go. Yep, that's perfect. Yep.
Sorry. Yeah, no, I really learned a lot, appreciated hearing all the speakers, but I wasn't prepared to ask questions as opposed to ask.
No worries. Didn't mean to put you on the spot. Just wanted to give you the opportunity.
Flexibility on my part.
No worries. Thank you. Other questions? Yep, we got a question over here. Hold on one second.
Hi, I'd like this to be a question for all of you. It kind of stems from some of the terms I've been hearing surrounding patient care, like average consumer, social context, alliance, and even comorbidities. In either of your research or your development, what are your considerations with like differences that come to race, ethnicity, and culture? Because I would imagine that those affect everything from genetics to direct patient care to looking at like your consumer market.
I can speak quickly to the genetics if that's okay.
Sure, please. Thanks.
One issue in the genetic studies is they've been largely or predominantly of European origin. And one thing we didn't talk about is the development in medicine of polygenic risk scores, where you take all these thousands of variants and come up with saying someone's at higher or lower risk, let's say for schizophrenia, where it works the best. And those are there are two problems. One is the data to drive that is almost all European and not generalizable. And the second part is, as you know, we look around and as is a good thing, people don't come from one of the three main populations, much less subpopulations of Asia, Africa, and Europe, Western Europe. So when we start to do those studies of polygenic scores, we'll often lose a lot of data because, parents may not be of just one population origin versus both. So that's actually a big challenge for the future, both to include more diversity in those samples as well as to develop models that can address the complexity and diversity that we live with and are glad to embrace.
Thanks, Ed. Chubao?
I think Dr. Cook. What Dr. Cook talked about is polygenic risk score, like from genetic perspective. From the the biology side, actually, when we try to use iPSC to model the disease risk factors, we also face the question, what kind of donor, like genetic background we want to use. Like, you only use one cell line or different cell line from different ethogenic background, or male and females. Those questions. For some highly penetrant genetic risk variant, for example, what we found, for example, the SETD1A gene I mentioned, we studied quite a few genetic background. We found, if you look at the biological effect, actually they're pretty similar actually for those highly penetrant genetic risk factors. But for common GWAS risk variant, we knew it's kind of population specific. There are a lot of loci. They are specific to different populations. Despite different population specific common risk variant, if they point to the same gene, you would also expect the same biological effect from the specific genes, right? So I think there are different ways to look at this issue, from biological perspective in the context of genetic association, I think.
All right. So I had a question pop up in the chat here from Dr. Noel. So I hope I'm getting this correctly. She was asking about the family of a patient on Clozaril, who was asking about the Lateman protocol and the Team Daniel protocol. I've not heard of either of those, so... Okay. Anyone in the audience? Yeah, so Amy, no one's aware of those. We'll try and get some more information and get back to you. Other questions? Oh, yep.
I am not a clinician, so I want to thank you all for what you do, and because I'm an outside person that has experience with family, I guess, having some of the issues. So I wanted to do that. I also wanted to say regarding the AI, I raised kids when the internet was starting to come out, and the instant messenger started. I had a son in middle school, and another mom said to me, "Isn't it great? They can just type on the computer and they don't have to face each other." I'm like, do you hear yourself? No, this is not a good thing. We need family, or we need people contact and stuff. So that's just a sidebar. My question is, have you found any methodology or suggestions when you have people that you care about that don't think they need help, or don't like the medical community, or don't think anything's wrong with them? How can you start to convince them? I don't know if with any of your experience, if you have any ideas.
Yeah, that's a really difficult situation. Yeah. I think there are maybe some different approaches. I don't know if there's a try do two approach. I think if there is someone that person does trust and can be a liaison between the medical community or a clinician and themselves, like a family member that they do trust the opinion of, or they do trust having kind of like an intervention, like, okay, let's all sit down and talk together type of thing. I mean, I've had some success with that. Or I guess kind of ramping up the external controls depending on the risk and severity, like family needs to set these boundaries now, and if you can't agree to continue doing treatment or continue taking medication, you can't stay with us anymore. I'm sorry. Like having to set some like tough love limits on things. Ultimately, there's a legal pathway. If someone is significantly impaired and they really need medical care, to go in front of a judge and have a judge make a determination that this person needs either inpatient care or like an outpatient medication order.
Yeah, and I'll just piggyback on that. I was looking around the room. I thought Dr. Goldenberg might be in the room. I hope he's listening online. One of the issues we've been struggling with a patient at Swedish recently is the conflicting ethical dilemmas of autonomy and paternalism, right? And how do we find the right balance in that to allow someone self-determination, making decisions, even if they are the bad decisions or decisions we might not agree with or make ourselves? Where does that line get crossed? Until then, it's an issue around safety and, risk, right? And that is a gray zone that, does not have clear parameters that direct us. So, it's a little bit of that feel of, wow, this feels like we're now getting into a situation where I need to maybe involve the legal, guidance and other things, because that is a constant dilemma that we face in psychiatry particularly.
I'd like to add something to the question of the book, I'm Not Sick, I Don't Need Help, by Javier Amador. I'd strongly recommend. It's helpful to families, has a lot of really good ideas about, I think the basic concept that we all use is what is important to the person. I think Dr. Marvin was alluding to that. What are your goals, without having to speak to diagnosis that may, as he mentioned, particularly early in the course or dependent even longer in the course, without insight, to talk about what do they want for themselves, and how, for example, medication adherence might help with that.
Yeah, thanks, Ed. As a child psychiatrist, I certainly use that aspect of motivational interviewing, right? Which is, okay, getting treatment for this disorder is not at the top of your list, But getting your parents off your back is your most important concern. How do we get your parents off your back? It's maybe to go to your appointments or take this medicine or whatever else, right? So trying to use that as the way to spin that engagement. So that's certainly an important part of it.
So my question, which piggybacks on what he just referred to, the literature, is how many of the people that are clinicians are aware of that work and are using what is called the LEAP method? Is that something that you train everybody at Endeavor with, or?
So not that specific technique, but I think principles of that technique are generally things that are common in most multidisciplinary training programs. So whether you're talking about medical schools, social work schools, psychology, that, have certainly some aspects of that we bring in. And I think, the book that Dr. Cook is referencing, synthesizes that and frames it a little bit more for families rather than for clinicians. But there are absolutely lots of those skills that are critically important and part of our training.
I'll also put a plug in for the mental health care directive that the state of Illinois has. So in Illinois, we can basically put together an advanced directive for mental health care. So it's a document that anybody can fill out, and it's like an advanced directive saying, if I should become ill or incapacitated, this is what I want to have happen and not happen to me. And it's all focused around behavioral health and mental health. So it's medications I want or not want, ECT, yes or no. If I become hospitalized and I need decisional support, this is the person that can make these decisions for me. And then if the patient does get hospitalized and they're deemed to be unable to make decisions for themselves, this document kicks in and all those predetermined decisions can be effective and really help them guide care. And it can be really a useful adjunct to care for patients who, when they're feeling well, are insightful and have, good decision making, and they have their best goals in mind. But when they become sick, they kind of lose those capacities and end up, and then in my experience, it's the patient who has a manic episode and then 3 months later, they realize, wow, look at all this stuff I've done. If I had just gotten started on this medication I know works for me 3 months ago, I would have been better. So it can really help in those situations where they know what works for them.
We had a question come online asking about if folks could expand on biggest concerns about uncertainty in government. So I'll look right at you, David, as I ask that question. You're among friends.
It's so hard. It's so hard to know because there doesn't seem to be a set of guiding principles. So how, again, I feel like I can't speak my ultimate truth on this, but yes.
So,
worst case, best case.
We're all looking to think the same thing.
I just think that there's.
Yeah, so maybe I'll start and then see if there's something you want to jump in there, because I think that point about uncertainty is certainly a huge part, right? There's a certain amount of predictability that goes on as administration changes, right? There's some Democratic Party principles, there's some Republican Party principles, and so you get some sense if, the ground shifts one way or the other way, we're going to be within the rules. We'll lean a little left, lean a little right, but we've got some predictability about that. So I think it seems like we've lost a lot of that predictability. So that's certainly one major uncertainty. I think the other piece of it is that it seems that you've got individuals making decisions based on their own personal life experiences as opposed to the evidence base. So I saw that in one of your first slides, right, was about evidence base being critical for some of the components. And without that evidence base, and it's just like, oh, Ramon, I decide I like, sunny days rather than rainy days, and all of a sudden we're out in left field.
The thing is, I'm trying to be sort of fair and impartial, which maybe is impossible. Look, you guys know the geography I live in, and you can guess at my politics. But I also want to say that when it comes to things like junk food, right? Like that might be a good thing that comes out of this. It might be coming from a person who is not of sound state and for reasons that we don't understand, but there might be some strange benefits that happen. I'm not banking on too many of them, but I will say that things that I think are relatively safe are Medicare. I think if you touch Medicare, it's a third rail, and my hope would be that nobody goes there. And if that happens, maybe the next election will be different. And when it comes to investing in novel drugs, I mean, and things like the DEA, I don't know. It's very hard for me to make sense of it.
Yeah, no, I think that unpredictability is certainly the biggest part.
And one concern I have is on the regulatory standpoint. I think one of the efforts is to decrease regulatory burden. And my concern about that, I guess, without evidence, I guess, is the risk for safety and safety events. If there's less regulatory burden, or less reporting required around certain things, that's going to open us up to more risk. We'll have to see on that one.
Fair enough. All right, and I think I saw the last question pop up there from Rena, which is for you, Ed, about the interface of OCD and spectrum, someone who gets stuck on almost every word and not in perfect alignment with obsessions. So wondering if you can think through that a little bit and feel comfortable replying.
Tough situation. Should I say what the question was?
If you can synthesize the question, I'd appreciate it.
Yeah, interface of OCD, also likely spectrum, which I assume is autism spectrum. Adult refusing treatment, gets stuck on almost every word, not in perfect alignment with obsessions. Believes irrational fear is true. Then goes down a rabbit hole with yet another word. Not available for reciprocal conversation. The only human connection is with family, so family maintains ties in a logical dialogue for reassurance. Difficult case. I could have given a talk or the whole morning on difficult cases.
That's for next year, Ed.
Yeah. One of the disadvantages of decades of treatment is talking to the parent about how you haven't really gotten that far in all this time. But, I mean, from what I'm hearing, trying to treat the OCD, which I assume, knowing how skilled Dr. Goodfriend-Levy is being done well. Hopefully able to get the family and the patient to comply with that. A lot of ideas about how to work on refractory OCD. And then the other thing that's alluded to here in OCD treatment especially is, of course, you want to do exposure with response prevention, but when the anxiety is so high and the internal motivation isn't high enough, it's hard to actually get past the just wanting to reduce the anxiety acutely, which is going to increase the anxiety and the connection between anxiety and compulsions over time. In families, it's hard. I wouldn't say that I would be good at this myself, but it's hard to work with the families not to, as Dr. Goodfriend-Levy mentions, sort of give in and reassure, which is sort of a natural reaction, which I think is some of the paradox of OCD behavioral treatment, that you're having to do what wouldn't come naturally to try to reduce anxiety.
Super, thanks. Let the microphone come to you there. And Zahava, we'll make this the last question here in the audience.
So I never really knew about the relationship between autistic spectrum disorders and psychotic disorders. So it sounds as though there's some genetic overlap. I wasn't clear on that. So anything you could say to elucidate would be helpful.
Yeah, I can say something because I was like, oh, why am I invited to this? I think it's because we're using similar tools. I did show, and Dr. Dwan showed overlaps in some of the rare findings. So, for example, if you have the 22q deletion, it's interesting. It often presents as autism or ADHD early, and then psychosis happens later, and consistent with the usual psychosis onset that Dr. Marvin said. In general, and that's the problem, saying in general, there's not an increased risk of psychosis in autism, and if there is, it's probably more mood-related psychosis. But I think the point is, in getting to these more specific causes, like the 22q11 deletion, or like orexin deletions, or the 16p11 duplications, it's not the overall 1% with schizophrenia and about 1% with autism and lumping everyone together, but trying to get much more specific diagnoses where if we did identify the 22q11 deletion as somebody presenting young with autism or ADHD, we would see them at high risk for psychosis.
Thank you.
Super. So appreciate everyone's engagement. I'm just going to see if anyone on the panel has any last thoughts before we wrap up. Lots of head shaking? No? All right. Well, thank you all. Yep. Have a great day and stay tuned for next year. Thanks, Ed.
Thank you.